Good morning, everyone. Welcome to the 2022 UKCPVS stakeholder meeting. My name is Catherine Harries, and I'd like to welcome you all here today. So just a bit of housekeeping to start off with. So everybody should be muted. We'll have the UKCPVS presentation first, and then a break around 11:35. And just to let you know, this webinar will be recorded and it will be made available after the event. Please complete the survey at the end so we can help improve our webinars in the future, and there's also a few questions in there about the UKCPVS project itself. So to ask a question, hopefully this slide is self-explanatory. Click the arrow in the orange box and then click the white arrow, and yeah, let us know if you've got any questions. And please ask questions throughout the presentations. We'll filter them out at the end, and there's a discussion session at the end as well. So this is the agenda. We have a good range of speakers here. We've got first the UKCPVS presentation from the 2021 season, and then we have Diane Saunders, Rachel Goddard, and Stuart Knight, and then the discussion at the end. And just a reminder here that any AHDB publications can be ordered from the website, so you can get the recommended list and pest and disease guides. And we have presentations and webinars on our YouTube channel, and this presentation will be uploaded to that after the event. Oh, sorry. And just a reminder that we AHDB have a Shape the Future vote where we're asking levy payers what they want us to look at in the future, and we'd be really grateful if you could sign up to that, and encourage any levy payers you know to sign up to that by the end of March at this link for the votes in April. So now I'm going to hand over to John Lucas, who's going to be chairing the event today. John Lucas is from Rothamsted Research. Thanks, John.
Thank you, Catherine. Just accessing my screen. I think we have to put this up a bit. For some reason, it's gone right to the end. Oh. No, I can't get to the end here.
Can you go through?
All right. Okay, sorry about that. For some reason, it opened it at the last slide. Okay, welcome everyone. It's self-evident, I think, that after the last 2 years, the need for disease surveillance is not really needed to be stressed. But of course, most people will perhaps not be aware that there are also threats to plant health in addition to human health. And here's one example: yellow rust on cereals, one of the diseases we'll be focusing on today. And of course, it's a moving target, just like COVID is a moving target. We get the emergence of new biotypes, including recombinant pathotypes that can threaten the resistance of current cultivars. In addition, we're not just trying to monitor one disease. There's a complex of diseases on crops, including cereals. 3 of them here: brown rust, Septoria tritici leaf blotch, and powdery mildew. But we now have a number of tools available for disease management. Agronomic, for instance, we can alter sowing dates. We know that plant nutrition is important in terms of disease susceptibility. But the 2 main tools that we use, particularly in cereals, are resistant varieties and fungicides. But of course, there are constraints on both due to pathogen evolution for virulence and fungicide resistance. Now, The good news is that plant breeders have worked hard to produce varieties that are less susceptible. And this is a comparison of the recommended list ratings for yellow rust 21 and 22. And this shows the number of varieties in each category. The good news is that the majority of them are on towards the right side of the screen, the more resistant categories. So there's a good choice there for growers. Of course, there is a proviso, which is the ratings can change due to changes in the pathogen population. In terms of fungicides, we've lost some of the most useful actives like the multi-site chlorothalonil and the azole epoxyconazole, but there are some new or reintroduced actives. We've got Folpet available as a multi-site, and we've got a new azole and a new target site, the QIIs, that are now available to growers. So there are some options. We know the story, of course, of what's happened to the azoles. This is actually Septoria and data from the AHDB fungicide efficacy trials over seasons from around 2001. And we can see there with prothioconazole, there's been a gradual loss in efficacy. But if you look at the far right, the new azole mefentrifluconazole is still showing good activity. So again, a moving picture. Now, until recently, the rust fungi were regarded as less likely to develop resistance to fungicides, so a lower risk. But we now know from experience, particularly in Latin America, with another rust, Phacopsera pachyrizae on soybeans, very damaging disease there, that over time, some fungicides have become less effective, in particular azoles and some of the excuse me, some of the other active groups as well. And a recent paper published actually from Diane Saunders' group, I'll just show the title here, has actually been looking for mutations in the azoles and STHI categories in yellow rust here. And I'm just going to read that final conclusion. These findings confirm that cereal rust species are not immune to fungicide resistance and indicate that appropriate screening regimes and resistant management strategies are needed. So I think the take-home message from all of this is that we need to be vigilant in terms of keeping an eye on the pathogen population for a number of reasons. And this is one of the main themes, obviously, of today's meeting. At this point, I'm going to conclude my talk and hand over to the RCPVS, UKCPVS stakeholder, UKCPVS team who have been carrying out the survey.
Thank you, John. Okay, good morning, everybody. My name's Charlotte Nellist, and I'm the project lead for the UKCPVS project. And myself, Amelia Hubbard and Sarah Wilderspin will be talking you through the results of the last year's survey. So this is just a quick overview of how the presentations will go. So I'll start with an introduction. Then Amelia will talk you through the wheat yellow rust work. Sarah will cover the wheat brown rust work and the wheat powdery mildew work, and then Amelia will cover the barley mildew work. And then it'll be back to me to talk a bit about the work we've been doing on genotyping wheat yellow rust. I'll briefly touch upon the Rustwatch project and sampling going forward into the 2022 season. So just a quick introduction to the programme. So for those of you who are not aware, the UKCPVS programme has been going since 1967, and this was following an outbreak of yellow rust on the previously resistant variety Rothwell Perdex. And the aim of the survey is to identify changes in pathogen populations and detect new races that may have an adverse effect on cereal production in the UK. The programme relies on yourselves, so farmers, agronomists, trial staff, breeders and researchers, to send in infected leaf samples. And this is so that we can see if there are new strains that are out there in the UK or ones that we already know about. And the programme is jointly funded by AHDB and AFA. Currently, we survey these pathogens. So we survey wheat yellow rust, which is also known as striped rust, or wheat. We also survey wheat brown rust or leaf rust, and then the two powdery mildews on wheat and barley. So I just want to talk you through the processes of what happens when we receive samples. So in this example, I'm just going to focus on the rusts. The process is slightly different for mildew samples. Unfortunately, we're not able to test every rust sample that comes in, but when we receive samples, they're registered on our database and they are placed into incubation chambers, into the incubators, where we bulk up the pustule should start to sporulate, and then those spores can be transferred onto a susceptible variety for multiplication. This takes about 14 days, and then we can collect those spores. However, if we need to bulk them up further, then this can take several weeks, where we can collect off the plant multiple times. And then we take these, the initial leaf samples are harvested and stored in -80 degrees, so that a selection of them can be genotyped later on. And at the end of the season, we select 25 isolates of wheat yellow rust and 25 isolates of wheat brown rust to go on to seedling differential tests. And I'll come back to that in some later slides. We aim to complete these differential tests by the end of December of that year, and then we select 5 of these isolates, which historically has been based on seedling differential tests, but we're now using genotyping information to feed into this, to select our 5 isolates, which will go on to adult plant tests and variety seedling tests. And these will be tested in the field the following year. All isolates, if multiplied successfully, are put into long-term storage. So we freeze dry these spores in sealed glass ampules, and they can remain viable for many years. Our yellow rust collection dates back to the early 1970s. So the aim of the programme is to identify changes in pathogen populations. This is done in a 2-step process. So we want to first identify the population change, and we do this through these differential tests. So on seedlings, I mentioned we screen 25 isolates in a normal year. We then go on to identify the risk associated with this change, and this is where we use the adult plant trials and the variety seedling tests, and 5 isolates go into these screens. We also use genotyping of a selection of isolates to help identify population changes. And the reason we do all this is because single major genes control varietal resistance to yellow rust and brown rust in wheat, and these can easily be overcome by new races in a short period of time. So this is the reason why we monitor it. And by tracking these changes, it gives us a first sign that the pathogens are evolving and adapting to the resistances that are in UK varieties. So just to give you a bit more details about the differential tests, each test contains differential varieties which have known resistance genes. So this example is for yellow rust, and we have these example differentials listed on the left-hand side here, and these carry these specific resistance genes. So if we look, for example, at Chinese 166, this carries the resistance gene for YR1. So if an isolate of yellow rust is able to infect this differential, it's said to carry the corresponding virulence gene for YR1, and it's able to overcome this overcome the resistance in the line. So we look very closely at the results to monitor the pathogen changes. Each isolate, when it arrives in the lab, is given a unique code. So this is made up of the year that the isolate is received and then the sequential number of the of when it's received that year. So for example, 16009 shown on the left, and then these along the top here we have all the differential, or an example set of all the differentials that we've used for screening, and they're scored on a scale of 0 to 4, where the values between 2.4 and 7 are borderline infections. Anything above 2.7 is a compatible interaction, and so the isolate is able to overcome the resistance in that line. Anything below 2.4, shown in green on here, is an incompatible reaction, and the isolate does not carry the associated virulence gene. So for example, here we can see that isolate 16184 didn't infect any of the YR7 differentials, and so is avirulent on YR7. Pathotype is another name for virulence profile of an isolate, and it will be mentioned numerous times throughout the presentation. So I just wanted to highlight that to you to ensure that that's understood. And the virulence profile of the pathotype is a list of the virulence genes that the isolate carries. And so we, as I've mentioned on the previous slides, we screen differentials that have known resistance genes, but we also have some specific lines that we don't know exactly which resistance genes they carry, but we know that they have distinct resistances. So we use these with their name. So the first part of it here are the virulence genes that have been identified, or the resistances that have been identified, and then the second part are these additional cultivars that we just code with letters. So we have rendezvous here, and Spalding's prolific, etc. All of this is detailed in our annual report, and so each race of yellow rust has its own unique particular pathotype. Our yellow rust race naming system combines genetic group and the pathotype. So we use a color coding system for the genetic group. So in this example, it's a red group, and then the race number is indicated by the number on the right hand side. So our genotyping was originally carried out by Diane Saunders at the John Innes Centre, but it's now being done in-house by Helen Bates and Anne Webbs, following Diane's protocols. So to identify risk in adult plant trials, we screen 5 representative races on RL varieties and candidates, and these trials are carried out in hand-sown tussock trials. And we run 5 individual trials for yellow rust and 5 individual trials for brown rust. These are inoculated with, each inoculated with a different isolate, and the picture shows how one of these trials is laid out. The trial has 2 replicates of each variety in there, randomised, and we erect this windbreak and netting around to help protect the plots and to maintain a mini climate within the trial to aid rust development. We disseminate results in a lot of places, so the slides are available in the handout. So if you want to find out more information, please do read our annual report. If you're attending the stakeholders meeting, we also disseminate results on the NIAB website and AHDB website. So please do have a look at all of these resources. I'd like to hand over now to Amelia Hubbard, who will talk you through wheat yellow rust results.
Good morning, everyone. So I'll be taking you through the wheat yellow rust report. So firstly, I'd like to point out the letters in the top right-hand corner. So WYR, this just stands for wheat yellow rust, and then when we move on to the brown rust, it will change to WBR, so you'll always know what disease we're currently talking about. So firstly, a little bit of background about the wheat yellow rust. We had the incursion of the warrior group in 2011, and that led to these highly diverse derivatives of this group. And it displaced our old UK population. So races like the solstice race and the robicus race, we just don't see those anymore. Then in 2019, we had more isolates identified carrying the virulence for YR8, Chronic and Crusoe. And over the years, we've seen the tumbling of resistance ratings for some of the RL varieties that are just under attack from these new strains. So I'd like to start by going back to the adult plant trials. So these are the ones that we were in the ground in 2021, and on the full set of RL and the RL candidate varieties. And these are the 5 isolates that we chose from the 2020 survey. So firstly, we chose 20050 from Cados Siskin. So back in 2020, we received 5 samples from Siskin. So we were keen to investigate those further at adult plant stage. We also chose, oh, sorry, I just changed my screen. Can you still see it? Oh, I'm back again. So then we chose 20092 from LG Astronomer. That one had virulence for Chronic, Apache and Evolution at seedling stage. And then we also chose 20191 from Cados Siskin. That one was particularly interesting for us because it had virulence for YR8. And then we chose 20293 and 20304. These were both from Cados days, and they were both infected Crusoe at seedling stage. So we chose both of those just to see if there would be any difference between them at adult plant stage. So here I'm going to present to you just some of the results from those adult plants. This is a percentage mean plot infection over 3 assessments. So first of all, we have robicus. That was infected in all 5 trials, and each colour represents a different isolate. So it had between 50 and 60% for those isolates. Then we have Cados kinetic. That one was more susceptible to 20050 and 20092 in the blue and the green colours. Next up, we have Cados Cyet. That one was also susceptible to all 5 isolates at around the 20 to 30% mark. And I've put up Cados Keren as well here because that had the same resistance rating as kinetic last year. And you can see that that one was more susceptible to 20050 and 20092 as well. The next varieties I'd like to show you are Ambition, Warrior, Cados Extase and Crusoe. So Ambition was particularly susceptible to 20293 and 20304 in the navy and the brown colour. It actually reached over 60% mean plot infection with 20304. Warrior was also susceptible to just those 2 isolates, much lower levels, but it was around the 10% mark for those 2 isolates, 293 and 304. And Cados Extase showed a similar pattern. So if you remember, Cados Extase is the host variety, the 20293 and 20304, and they were both able to infect Cados Extase at adult plant stage. And finally, we have Crusoe. So you can see that one was really only susceptible to 2304, which was a little bit of a surprise to us. We did expect 2293 to be to show some reaction on Crusoe, but it was just 304, which basically just shows there is a difference between those two isolates. The last set of varieties I'd like to show you are Torch, SY Institute, Chronic and Solstice. So you can see Torch is also showing a similar pattern to the varieties we've just looked at, where it is particularly susceptible to 2293 and 2304. And it actually reached over 60% for both of those isolates. SY in situ, I picked this one out as well because it shows this same sort of pattern with those two isolates in the navy blue and the brown. So it's just interesting that it does show that there's a clear difference between our trials. And again with Chronic, that's one of our control varieties. Again, it was more susceptible with 2293 and 2304. And I just popped up on Solstice here just as a compare it with those results with a susceptible variety. And you can see again, Solstice, we know it's susceptible, but it was actually more susceptible with 2304. So now we can have a look at the varieties that were resistant to all 5 isolates. And I've taken this as just listed the ones that had 2% or less average infection across those 3 assessments. And there's 26 varieties up here. I've got Rendezvous in the darker colour just because that's one of our controls. And you can see we have really good varieties here. These plots are absolutely blasted with inoculum during the season. I inoculated them about 10 times. So they were under heavy disease pressure and they still had less than 2% infection across the season. There are 4 standout varieties, which I've put an asterisk next to, which is Caduus Guium, Caduus Palladium, LG Farrier, and Theodore, because those 4 varieties didn't even have a hint of yellow rust. So not even a single stripe or a few pustules, just nothing. They were completely clean, so they did really well. So just to summarise those adult plant trials, we had good levels of infection achieved, which just proves our inoculations were successful. Results confirm 2293 is a warrior. It's a pink type isolate that's been shown in our seedling tests, and it's also been confirmed as a warrior isolate by SSR genotyping analysis conducted by the Global Rust Reference Centre. 2304, that was very similar at seedling stage and at adult plant stage, but it's actually been identified as a red isolate. Some varieties were clearly more susceptible or resistant to different isolates, such as Caduus extase, SY in situ, and Torch were more susceptible to 2293 and 2304. And as we've just seen, many of the recommended lists of varieties that are on the list for last year were resistant to all 5 isolates. So as well as our adult plant trials, we also do variety seedling tests. So these mirror the adult plant trials. They contain the full set of RL and RL candidate varieties. Here I've just popped up some of the results and we have the seedling reaction on the left alongside the adult plant reaction on the right. So the seedling is a 0 to 4 scale, as Charlotte's just shown you, and the adult plant scale is a 0 to 100% plot infection. We take the top 3 varieties. We've got Theodore, Caduus Siskin, and LG Astronomer. So you can see by the green colour that these were resistant at seedling stage and they were resistant at adult plant stage. So they have good overall resistance. Next up, I have Illicit, Elation, Caduus Jackal, and LG Skyscraper. So these 4 varieties were susceptible to 2 or more of the isolates at seedling stage, but they were resistant at adult plant stage. So this means that they have this adult plant resistance that kicks in at later growth stages. Next up, I've put Crusoe, Caduus extase, SY in situ, Chronic, and Ambition. So these were susceptible to 1 or more of the isolates at seedling stage, but they were susceptible to 1 or 2 of the isolates at adult plant stage. And then we have Skyfall, we have Zayat, Torch, Virulence Kinetic, and our controls Solstice and Robigus. So these ones are all susceptible at seedling stage. Perhaps Torch has got a slight lower value there with 20,050, but these were all susceptible at adult plant stage, with the exception of Torch, which I must admit does stand out like a sore thumb on my table, but that's because these varieties have been ranked in order of their adult plant reaction. So because Torch had these exceptionally high levels on 2293 and 2304, it actually falls below Zayat in the table here. So now I can move on to our 2021 samples. So this map shows the location of where those samples came from. We received 155 samples. They came from 19 different counties and across 54 different varieties. So the majority of the samples did come from Lincolnshire, but we also had from Cambridgeshire and Essex. And we also had samples from Scotland and the Southwest. So we had a good geographical spread of samples. So I'd just like to say thank you to everyone who did send us in samples. And this slide shows the host varieties of those 155 samples. So we've got the 54 different varieties here, and I've just colour coded them in terms of the number of samples that we did receive. So Cadus Firefly was the most heavily sampled variety. We had 14 samples in from that one. Gleam and Skyfall in purple, we had between 11 and 13 samples in. Then we have the varieties that were in pink, so between 5 and 6 samples from varieties like Graham and Cadus Kinetic. We had between 3 and 4 samples in from the varieties in blue, so we had 4 samples in from Siskin. And then we had the varieties in the orange colour, we had 2 samples in from them. And you might notice some older varieties like Brimstone and Parade there. They have come in from our trap nurseries, or they may have come in from research plots. And then we have the varieties in green. We only had 1 sample in from those, so varieties like Illicit, LG Illuminate and Crusoe, we only had 1 sample in from. So if you've joined us for the last few years, you'll realise that 155 samples is actually fewer samples than we have had in the last couple of years. And I think we can have a look at the cereal disease season to somewhat explain why that might be. So we had this wet autumn in 2020, but overall it was a mild winter. And then yellow rust did actually start to develop in March. And then we had a very dry April and temperatures were 2 degrees below average. Then we had lots of rains and storms in May, and that affected disease development in some areas. And it led to this very short, sharp disease season with yellow rust rapidly drying up and dying off by the end of June. And as a result, sample numbers were much lower in 2021 compared to 2020 for both of the wheat rusts. And I thought it might be actually quite interesting to have a look at the sort of like the sample intake. So if we actually pop back to 2020, 2020 was a bumpy year for yellow rust. We had 306 samples, but this graph just shows the sort of like the normal pattern of how samples come into us. So they start coming in March, and then things start to ramp up in April and May, and June is always our busiest month. Now, if we compare that to what happened in 2021, you can see that, yeah, March actually was looking really good for us. We had over double the amount of samples in March, but then April, things kind of tailed off, and we didn't see that our usual sort of influx of samples in April. Then there was a big drop in the number of samples we received in May. And then when the weather conditions were a bit more favourable, we saw an increase in sample numbers. So we had 78 samples in June. And as I've just mentioned, the weather could be responsible for some of this. So I thought we'd have a look at the weather. So this is a UK mean temperature, and I've taken it from October right through to July, because basically anything that comes in the autumn from sown crops we class that gets added onto the survey for the following year. So as you can see, the temperature in January was actually almost 4 degrees colder than it was the previous year. But we also had this very cold April and May. And what this lower temperature does, it increases the latent period. So the latent period is the date between infection and sporulation. And the colder it is, the longer that latent period will be. So I think that basically delayed our epidemic last year. And then if we have a look at the UK mean rainfall, you can see that, we had quite a wet winter, but April was quite dry, and then we had these rains and storms in May. And yellow rust, yes, it does like it humid, it does like it wet, but the trouble is with such heavy rain, it ends up literally washing off the spores from the leaves, so they can't go anywhere, they just land on the ground. So I think this has basically shown why we had these fewer samples in 2021. So our wheat yellow rust season highlights. We had 155 samples, and the majority of those did come from Lincolnshire. And despite receiving fewer samples than in recent years, 54 varieties were represented in the samples we received. The most sampled variety was Cadbury's Firefly, which was rated a 7 last year. And we had 4 samples in from Cadbury's Siskin, rated a 9, and none of those have reinfected siskin at seedling stage. We did have some unusual sightings and reports. One of these was regarding the Cadbury's Siskin sample, and we also had a report of yellow rust being particularly difficult to control on Cadbury's Kinetic, and samples from both of those were received and tested. So we're actually funded to test 50 rust isolates, wheat rust isolates, and because we actually had lower brown rust samples in last year, we actually ended up testing 40 wheat yellow rust isolates. So we can have a look at the host varieties of those 40 isolates that we tested. I've colour-coded them in terms of their resistance rating. So you can see we tested 4 isolates from Graham, 4 from KWX-Stays, 4 from LG Skyscraper, and 4 from Skyfall. And then we tested 3 from Siskin and 3 from Wolverine, and 1 sample from the rest of the varieties listed there. And you can see from the colours that we didn't just focus on testing samples or isolates from varieties with high disease resistance ratings. They are useful to us because a highly rated variety with yellow rust might be the first sign of a resistance breaking down, or perhaps a new pathotype. But it's the susceptible varieties like Kerin and Kinetic and Skyfall, which actually they kind of like act as a rust sponge. They will suck in any yellow rust that's in the area. So they are good indicators of the race that's prevalent in local areas. So now I'd like to go through the seedling virulence test in terms of the virulence frequency. And this is a graph showing the percentage of isolates carrying virulence for the resistance genes. So virulence for YR 1, 2, 3, 4, 6, 7, 9, 17, 25, and 32 all remained very high in the population. We did see a little bit of a dip in virulence for YR3 and YR4, but nothing too unusual. But it was YR8 that we were keeping a close eye on, because that did see a peak in 2019 when it reached 15% of the samples, and in 2021 it was seen in 8% of the samples. So it's still remaining at fairly low levels in our population. No virulence was detected for YR 5, 10, 15 or 24. So if you have a look at that graph, you may think, well, that kind of like shows a rather sort of like stable population for the wheat yellow rust. But it's not until we look at the additional cultivars that it's a little bit of a different picture. So I've only included some of them on here. We do have quite a few extra varieties in our seedling test. So we include things like Sterling and Warrior. We also have some of the interesting RL varieties, like we've got Siskin in there, we've got Extase in there and Costello. So I've just picked out just some of the ones to show you today. So Virulence for cadenza, that was seen in almost 90% of the isolates that we tested. Virulence for Apache, that actually peaked and was seen in every isolate we tested, so 100%. Virulence for evolution there, so that one has actually fluctuated the most, and it was seen in 38% of the isolates that we tested in 2021, but it's on a par with its 2018 level. Virulence for chronic, that one has been increasing over the last few years, and it was seen in over 40% of the isolates that we tested. And virulence for Crusoe has remained quite low in the population, and we only detected it in 3% of the isolates that we tested. So the common pathotypes that we found in 2021. So we found 3 common pathotypes. They were red 36, red 37, and red 41. You can see that they have quite similar pathotypes, with perhaps red 41 being more simple. It doesn't have virulence for rendezvous. And red 36 and red 41, they have both been seen before. They were detected in 2019. And red 37 was actually the most common pathotype that we detected in 2020. So there was nothing new to report there. We did detect some new pathotypes though in 2021. We had 7 new pathotypes. So the top one is quite interesting because it had this A virulence for YR3, YR4, and it didn't infect rendezvous. And then the one at the bottom is particularly interesting because it had virulence for YR8 and combined that with virulence for Crusoe. So we quite often get asked about whether there's any correlation between the location from where these isolates were sampled from and the distribution of those new pathotypes. So we had one new pathotype coming from Kedibus jackal, and that's the one I've just shown you with the virulence for YR8 and Crusoe. And it came from Lincolnshire. We had this one here from Lincolnshire, from RGT illustrious, and we also had this new pathotype coming from a sample from Barrel from Lincolnshire. So that's 3 out of the 7 new pathotypes that did come from Lincolnshire, but Lincolnshire was the main area that we had received samples from, so I don't think it's surprising that we did see quite a few new pathotypes coming from there. So, but it does show how important it is that we have a wide range of samples to coming into us, because it just means we can cast our net further and pick up any changes in the population more easily. So the other new pathotypes, we had this one here from Kedibus extase. That's that simple pathotype with a virulence for YR3 and 4. That one came from Norfolk. We had a new pathotype coming in from Crusoe from Oxfordshire, and we had Kedibus kinetic also gave us a new pathotype from Herefordshire, and Kedibus siskin gave us our final new pathotype from Warwickshire. So we also get asked about whether there's any difference between the pathotypes we see at the beginning of the season to the ones that we see at the end of the season. I just thought I'd show you that we tested 21003 from LG Skyscraper from Oxfordshire, and it had this big broad pathotype. And we also tested one of the last samples that came in, which was 21154 from Kedibus extase from Norfolk. That's that simple pathotype with the A virulence for YR3 and 4. And you can see there is quite a difference between the pathotypes there, but we do see different pathotypes throughout the disease season. So it's not something that we would actually think is unusual. So if you joined us for last year, you may remember that Kedibus firefly is resistant in our seedling test. So I thought I'd just go over this again, because I did conduct seedling tests back in 2020 using some firefly isolates, And even though it's resistant in my seedling test, I can get good infection levels in separate tests. So we were thinking that it might be environmentally sensitive, and I wanted to do some further work, trial maybe some different lighting and different temperature regimes, just to see if there would be a difference. So I did the different dot lighting, and those tests, basically it didn't work very well because it didn't like the lighting that I tried. But I did try some seedling tests conducted at a reduced temperature, and I chose 12 degrees during the day, 10 degrees at night, using 2 isolates from the 2021 survey. So our normal seedling tests are conducted at 18 during the day and 11 at night with a 16-hour photo period. But I did see much better results at this lower temperature for K-Burst firefly, and I did see some nice sporulation going on. So yeah, we've seen this better sporulation on K-Burst firefly with 2 isolates from the 2021 survey at this reduced temperature regime of 10. And we actually received more samples from K-Burst firefly in the cooler months from March to April in 2020 and in 2021. So is this a coincidence, or could seedlings of K-Burst be more vulnerable to yellow rust at cooler temperatures? And yet that's what I've concluded, that it definitely does have this environmental reaction going on, and that's why it's just not being infected in my seedling tests. So I'd like to look ahead to our 2022 season. So we have picked our isolates that we're going to use in our adult plant trials. We've chosen 2112. This one is from K-Burst chillum, so that's a spring wheat, and it was chosen because it's got a pathotype for red 36, and that was one of our common pathotypes. We chose 21014 from LG Skyscraper. That's another one of our common pathotypes for red 41. We chose 21045 from Skyfall. So this one was particularly interesting because it has virulence for YR8. And not only did it have infected our Avocet YR8 differential, but it also infected Compare, and not all of the pathotypes that we see that infect YR8 infect both of them, but this one did. Compare does have additional resistance in its background, but we picked that one out of our chosen YR8 isolates. We also picked this one for 21102 from K-Burst Jackal, because that's that interesting pathotype with the virulence for YR8 and Crusoe. And finally, we picked 21135 from RGT Wolverine. That one, it's a pathotype belonging to red 11, but it was one of the isolates that I was actually using in my K-Burst firefly test. So I wanted to see how that one would perform at adult plant level. So now we can look ahead to the RL list for 2022-2023. So what have we got out there in the ground now? So as you can see by the darker green colours, we have a lot of the varieties on the current RL do have these high resistance ratings of 8 or 9. So with the varieties rated 9, we have the big names like Crusoe, Siskin, Costello and Theodore, but we also have some of the ones that were candidates last year. So the newly recommended varieties, K-Burst Palladium, K-Burst Guiam, K-Burst Briam, K-Burst Dorsum, Mayflower and LG Typhoon. And all those varieties actually did perform very well in our own adult plant trials. Our adult plant trials don't actually feed into the RL ratings, but it's just nice to see that our trial data did match the ratings for these varieties. With ratings of 8, we have RGT Illustrious, we have Elation and Illicit, we have K-Burst Extase, K-Burst Jackal, K-Burst Cranium, LG Astronomer, LG Print, Merit and RGT Saki, and joining them are the newly recommended varieties RGT Rashid and Champion. So with the varieties rated 7, we have RGT Barstow and RGT Stokes, and they are joining LG Illuminate, LG Skyscraper and Graham. So with a rating of 7, yet you may see yellow rust on these, but it should be at fairly sort of like moderate to low levels. With a rating of 6, we've got Caderus Firefly, so that's dropped a point from last year. We've got Caderus Barrel, Swallow, and RGT Gravity. With ratings of 5, we've got LG Spotlight, SY Incital, and Glean. And then with ratings of 4 this year, we have Caderus Kerin, Caderus Science in there, and RGT Wolverine. And then with a rating of 3, we just have the popular red wheat Skyfall. So I just thought I'd have a look at the variety rating changes over time, because my very first slide mentioned this tumbling of resistance ratings. So I just thought I'd compare how our varieties have held up over the years. So firstly, we've got Glean, that started life as a 7 and it's now rated a 5 going into this year. So you can see I have actually gone to the 1 decimal place here with these figures, but you may be more familiar with seeing it as a whole number. So Caderus Barrel, that one started life as an 8, and it actually withheld its resistance for 5 years, and it's only recently dropped down to a 6 for the 2022. Caderus Firefly, again, that was big news when it joined the recommended list in 2019. It was an 8.7, which would have been classed as a 9. And now it's rated a 6. Caderus Kerin and Caderus Science, so they both started life as a 7 and then now both rated a 4. We have LG Spotlight and RGT Gravity. Again, they were both rated an 8 when they joined the recommended list, and now they're rated at a 4.6 and a 5.8 respectively. So Skyfall. Skyfall has been on the recommended list the longest out of these particular varieties, and you can see it did really well for 5 years. It withheld its resistance rating of a 6, and again, it's only in the last couple of years it's dropped down to a 3. And finally, on my little slide, we have SY Incital. So that one started life as a 6.7, so it would have been 7, and it's now 4.9. So my wheat yellow rust summary is we received 155 samples in 2021. We had 40 isolates that were tested at seedling virulence stage, and 8% of those isolates were identified for virulence for YR8. 3% of the isolates carried virulence for Crusoe, and the most common pathotypes that we saw in our 2021 samples were red 36, red 37, and red 41. And we had 7 new pathotypes. 1 had virulence for YR8 and Crusoe. So I'd now like to pass you back to back to Sarah Wildspin, who will take you through the wheat brown rust.
Sarah, you're muted. We still can't hear you, Sarah. Are you muted now? We still can't hear you. Can we move on to Amelia again? We can't hear you still, Sarah. Yeah, can we move over to Amelia again, and then we'll sort out your issues and then come back to you.
So I'm just going to have to find my place in the slide now. Won't be a minute. So we're going to start with the barley powdery mildew while Sarah gets her sound. Okay, so with the barley powdery mildew, in 2021 we received 15 samples, and they came from 4 counties and 9 varieties. So you can see the majority of the samples did come from Lincolnshire again, and we actually had 6 unknown varieties. So that's just where the sampler hasn't given us all the information. So it just shows how important it is if you can include all your sampling details, but host variety and location is particularly important to us. And I'll just wait for a minute because my slide has disappeared, but you might be able to still see it. I'm back again. So the current barley powdery mildew differential set consists of quite a few old varieties that you may not have heard of. So before, when we were talking about the yellow rust, we had YR genes. Now we're talking about ML genes with the barley resistance genes. And we did include some older varieties in there, and we also have some susceptible controls, Funky and Bazooka, from the current RL list. So our barley powdery mildew tests, they are done on detached leaf segments inoculated with a single isolate, and they consist of 4 reps per test. So the picture just shows an example of just one of those reps. So each leaf in there is a different variety, and we would have 4 boxes like that to complete the test. They are scored on a 0 to 4 scale, where 0 is no sporulation, no mycelium, and 4 is abundant sporulation, abundant mycelium. And anything that scores 2.7 and over is classed as susceptible. So I haven't put on all the results for the barley powdery mildew. This is a 5-year summary, and like the yellow rust, it's percentage virulence frequency with just some of the resistance genes. So virulence for MLAB, that reached at 50% in 2021 isolates. Virulence for MLK1, that was had a sort of slight increase on the previous year. Virulence for MLA1 also reached 50% of isolates. Virulence for MLA3 was seen in 27% of our isolates, and virulence for MLA7 was seen in 58% of the isolates. So we also have the resistance genes for MLO11 carried by Apex. That hasn't been seen in any of the isolates we tested in 2021. Virulence for MLO11 carried by Riviera. That one hasn't been seen since 2017, so that just shows that the MLO resistance in barley is still effective. Virulence for MLA9, that wasn't detected in 2019 nor in 2020, but it was seen in 2021 at levels similar to 2017 and 2018. And there was no virulence detected for MLA13 for the 2nd year running. So we don't conduct any adult plant trials for the barley powdery mildew, so we just have the seedling test results. And this is the summary. We have 26 single pustule isolates tested from 15 samples. Virulence for MLH, MLRA, MLG, MLCP, MLA6 and MLA12 was found in over 80% of isolates. And virulence for MLA9 rose from 0 in 2019 and 2020 to 15% in 2021, which reached a level similar to that seen in 2018. No virulence was detected for MLO11 carried by Apex, MLO11 carried by Riviera, nor for MLA13. And that ends my slide, so I'll just try and pass you back to Sarah Wildspin to see if she can show you her presentation slides.
Hi Sarah, we still can't hear you. You are unmuted, but I don't understand why we can't hear you still. Do you want to just unmute and mute again? Just to see if that works. Are all your mics on? Okay, we still can't hear you. If we can move back to Charlotte and then we can come back to you, is that okay?
Hi again. Okay, yeah, my screen thing shared, sorry. Right, sorry, bit disjointed now. So I'm going to talk you through some of the genotyping work that we've been doing on yellow rust. So the aim of this part of the work was to develop routine genotyping of wheat yellow rust isolates using results from Diane's field pathogenomics project. And this is to categorize isolates into the different genetic groups. So over the last few years, we've been focusing on 24 isolates from the 2019 survey, 24 isolates from the 2020 survey, and then 48 isolates from the 2021 survey. So this is just a simplified diagram of the process. So we're following Diane's MARPL pipeline, so the mobile and real-time plant disease diagnostics pipeline, and DNA is extracted from the infected leaf tissue that's sent in from yourselves, and we use PCR to amplify 242 genes which are diverse between the different isolates, and these are pulled and then the libraries are sequenced on a gridiron, and this is a it uses long read sequencing technology, so we can cover the whole of these 242 genes. And then we run population genetic analyses, and this is to determine the relationship between isolates and generate a phylogenetic tree, which should look something like this. So this is our data today. I'm really sorry that I can't present to you today on a full screen where we can zoom in on this, and I appreciate that on some of your screens this might be really small, but I just want to talk you through the slides. So in the center of the circle, we have the phylogenetic tree, and this is the individual isolates from the survey, so those ones that I mentioned before, and also reference isolates that are known from the different genetic groups. And then around the outside, we have a heat map, and this is showing the pathotype data from the different isolates. So We've used colours on the genetic group. So again, looking at the centre of the tree, we can see this big band of red isolates, and this is the majority of the UK isolates that we've genotyped to date belong to this red group, and these have dominated for the last 3 years. So as Amelia mentioned earlier, we had 155 isolates that were sent in this year, of which we genotyped 48 of these isolates, and all belong to the red group. And within this red group, we see a broad range of virulence profiles. So in this heat map, if you look on the band on the outside, anything that is coloured red is is where the isolate is virulent on the individual differential. So as going out from the centre of the circle is the different differentials. So so red means the isolate was able to cause disease, so it's virulent on that differential. Green is an incompatible interaction, so the isolate was not able to cause disease. And then grey are isolates that weren't tested on those differentials. And so, yeah, in this red group, we see a broad range of virulence profiles. If we just focus in on the other part of the tree, so the genotyping has helped to identify unusual isolates, and we can see that in 2019, I hope you can see my pointer, we had 2 isolates that belong to the pink group. So this is the warrior group, and we also had an isolate in the purple group, which was the chronic group. And in 2020 we had these 2 isolates in the pink group. So this work is ongoing. We will genotype some more isolates this year and expand this analysis. What I should have mentioned is all of these isolates, as Amelia alluded to, we've selected isolates that have come from multiple locations and multiple host varieties, but we will add this into our figure to have a look if any of that is correlated. So I hope in the annual report we'll have more information to present to you, and also at next year's stakeholders meeting we'll have an update with this coming year's data. Organisers, would you like me to carry on and talk about Rustwatch, or do you think the issues with Sarah's microphone are fixed?
I think
if you just want to carry on.
Yeah.
So shall I stop here? Sorry.
Sarah's not back yet, so carry on.
Oh, Sarah's not back yet. Okay, well, I'll just briefly talk about Rustwatch then. Maybe you can just give me a shout if you want. I've got a sampling section after the next 4 slides, so you could give me a shout if you want to intervene for Sarah's section. Okay, so Rustwatch is a European project. It's a 4-year project with 25 partners across Europe. And it's building upon national programs such as the UKCPVS program, and it's focused on all rust pathogens, so yellow rust, brown rust and stem rust. It is a combination of people that work in VCU, breeders, ag chems and extension people, and the project is studying a panel of over 200 European wheat varieties and their resistance to rusts. So on the slide I listed the 5 work packages, and I believe over the previous years you've had some presentations where you've had people talk about this before, but for this presentation I just wanted to highlight the first 2 work packages. So the first one is understanding pathogen biology and drivers, and this work package is focusing on developing faster and more efficient diagnostic methods to track new rust races and genotypes. And we have submitted some of our data, as Amelia has mentioned, so some of the isolates have been sent for pathotyping and genotyping to our European colleagues, and this information is displayed on this Rustwatch Rust toolbox, and the link is at the bottom of the slide. And it's a really interesting tool because we can see which races are prevalent in other countries compared to what we're seeing in the UK. So if you have time, I recommend having a look at that resource. And then I just wanted to highlight some of the work that's going on at NIAB on work package 2. So this work package is looking at disease prevention by host resistance. And the aim of this work package is to use microphenotypes. So these are assessments that are done through the microscope to distinguish between resistant mechanisms in approximately 200 wheat varieties. And the assessments are classifying yellow rust growth stages into 6 classes. So we've got first one, which is just a germinated spore. The second one, where we see the germ tube enter the stomata and form substomatal vesicles. The third classification is the formation of infection hyphae and haustorial mother cells. And the fourth classification is the formation of the first runner hyphae through the leaf. The fifth one is the runner hyphae elongating through the leaf and infecting host cells. And then classification 6 is the formation of a pustule, as shown in the image on the bottom right. So just to explain these classifications a bit further, on the left-hand side, we've got growth stage 2. So at the top, you can see this germinated spore, and it enters the substomatal cavity to form this substomatal vesicle. And then on the bottom of the slide, we've got growth stage 4, where we see the formation of the first runner hyphae. And then we see this runner hyphae start to elongate on the right-hand side of the slide. And this is where it's then going to infect other host cells. And this work, the team have inoculated 204 cultivars, and they've taken samples for 5 different time points. And they've taken 3 leaves for 5 different time points. So they have over 3,000 samples that they are working through to classify into those 6 different classifications. So next year, hopefully we'll have the results that we'll be able to present to you about that. I'm now going to move on to sampling, unless Sarah's ready.
Yeah, Sarah's ready. I'll put on. I'll try her.
Okay.
Hi everyone, sorry for that technical glitch. Hopefully we're okay to go now. So hi, I'm Sarah, and I'm going to present the wheat brown rust and wheat mildew results for 2021. So a little bit of background information on wheat brown rust. The surveillance started later than other cereal diseases in 1973. At the start of the survey, there were limited options for, there were limited options. At the start of the survey, there was limited options for resistant varieties. To brown rust, for example, Clement. And in 2014, the brown rust population overcame the moderate resistance in Crusoe. So following on from when we left off at last year's stakeholders event in March 2021, we had selected the 2020 isolates to be tested at adult plant stage, which we then went on to inoculate and assess in the summer. So those adult plant trials were tested on recommended lists and candidate varieties, and here are those 5 2020 isolates. So 20002 was selected because it was a new pathotype in 2020. 20005 was selected because it came in on the variety KWS Firefly. It was a new pathotype and carried virulence for LR28. 20018 was selected because it came in on the variety Crusoe. It was a new pathotype and had the most complex virulence profile for 2020. 2001 was selected because it was the most dominant pathotype in 2020 and was the second most dominant pathotype in 2019. And 2032 was selected because it was the most dominant pathotype in 2018 and present in 2019 and 2020. So here are just a selection of a few of those adult plant trial results. The full results are in the UKCPVS report for 2021. So this graph shows we've got the variety along the bottom, and we've got the mean percentage plot infection across 3 weekly assessments for both reps on the y-axis. Above each variety, we've got a number, which is the 2021-22 RL rating, and each colour bar denotes one of the isolates that were tested. So for the variety Crusoe, it scored one of the highest scored varieties in our trial, and was classified as susceptible for all 5 isolates. And KWS Graham had one of the lowest RL ratings across the trial and also scored quite highly. LG Astronomer had one of the highest RL ratings and was classified as resistant to 3 of the isolates. And Skyfall and Theodore also had quite low RL ratings and was classified as resistant to all 5 isolates in the trial. I'd just like to note that the UKCPVS data doesn't feed into any of the RL rating data. I'm just using this as a comparison. So the varieties that are going to pop up on this slide I selected them out for what we saw in our adult plant trials was later seen in the change in RL rating. So we've got the 21-22 RL rating number on the left and the 22-23 RL rating on the right. So LG Astronomer was moderately resistant to 2 of the isolates tested, which isn't necessarily what we'd expect for an RL rating of a 9, and that then dropped to an 8. For LG Illuminate, it was moderately susceptible to 4 of the isolates tested. No, moderately susceptible, yeah, to 4 of the isolates tested, and its rating dropped to a 7. For LG Prints, all of the 5 isolates were. It was moderately susceptible to all of the 5 isolates, and its RL rating dropped to a 7. And for Theodore, it was resistant to all 5 isolates tested, and its RL rating increased to an 8. So all of the results for the adult plant trial will be presented are in the UKCPVS report for 2021. So moving on to the variety seedling test results. These seedling test results were inoculated with those same 5 2020 isolates as those inoculated in the field on the same set of varieties in environmentally controlled growth rooms. So I've just got here a snapshot of what's seen in the UKCPVS report. These are in table 13, and I've just highlighted the isolates which were resistant, the varieties which were resistant to all 5 isolates. Which I have then written out here for reference. So there were 15 RL varieties which were resistant to all 5 isolates tested. And then all other RL varieties were susceptible to all 5 isolates, except for LG Farrier and RG2 Bearstay. So if you see any brown rust at seeding stage, particularly on these resistant varieties, then please just pop a sample to us in the post. I'm now going to look at the variety seedling and adult plant test results. Again, these are in this table 13 in the UKCPVS report. And I've just selected here a few of those to talk through. So we've got the seedling results on the left and the adult plant results on the right, where green denotes resistant and red is susceptible, and orange is moderately susceptible and yellow is moderately resistant. Just take a look at Theodore. We can see it was resistant at seedling stage and at adult plant stage. And Glasgow and Skyfall were susceptible in our seedling test, but was resistant in our adult plant test. The resistance kicking in there. Whereas K-Doric stays and RGT gravity, we saw again was susceptible at seedling stage, and we saw a bit more mixed reaction at adult plant stage, but was generally moderately susceptible or susceptible. And Crusoe and KWS Graham were susceptible at seedling stage and at adult plant stage. So we can't necessarily make predictions from what's happening at seedling stage to what we're going to see at adult plant stage. So to summarise the wheat brown rust for 2020, for the variety seedling test, 15 RL varieties were resistant to all 5 isolates tested. All other RL varieties were susceptible to all 5 isolates, with the exception of algae farrier and RG2 bairstow. And for the adult plant trials, there was overall higher plot infection levels compared to last year. The results mostly reflected changes in RL ratings from 21-22 to 22-23, and there were no major changes in varietal performance that were reported. Moving on to the 2021 samples. In 2021, we received 10 samples from 4 counties and 7 varieties, and 8 samples arrived on RL varieties and candidates. The other one was from last year's RL ratings, and the one after was from an older RL rating. So most of the samples came in from Cambridgeshire or Oxfordshire, with there being overall quite low levels of brown rust in the season, which was represented in the lower amount of samples we received. What's the possible reason for this lower amount of samples? Well, as Amelia highlighted and presented earlier, it's very well or most likely linked to the weather seen in that season. So we had quite a few wet months, some late cold spells, and very limited hot sunny spells, which neither of those are really favourable conditions for brown rust. Here is a timescale of samples received in 2020 and 2021. In 2020, we had a few samples received at the start of June, but most of them came in at the end of June and at the start of July, and then started towards the end of July. And in 2021, we saw the lower amount of samples, with 5 coming in December, 2 in June, and 3 in July. So here is one of the first samples that came in, which on the 3rd of December on KW was carrying from Oxfordshire, and its pathotype. And here is the last sample, which came in on the 7th of July on variety Crusoe from Cambridgeshire. And here I've just highlighted the differences between their 2 pathotypes. So one of the first samples had virulence for these resistant genes, whereas the last sample didn't, and the last sample's pathotype virulence for the first one didn't, and the last sample pathotype turned out to be the most dominant pathotype for 2021. So here are the samples received recommended list ratings for 2021-22. We had 1 sample in with an RL rating of 3, and none of the samples received had an RL rating of 4 or an 8. And then most samples which came in had an RL rating of a 5, followed by a 6, and 1 had a 7. And then 2 of them samples were not on the 21-22 RL list. So from those 10 samples that came in, they were tested on our differential set at seedling stage to determine their pathotype. This graph shows the percentage of those isolates carrying virulence across the resistant genes, which will pop up along at the bottom over a 5-year period. So for the virulence for LR1, it's remained at 100% for the last 2 years. Virulence for LR3A has dropped a bit compared to last year. Virulence for LR17B has remained at 100% for the last 4 years. And virulence for LR20 has increased for the first time, whereas it's been dropping quite significantly over the last 4 years. Virulence for LR24 has remained at 0%, so none of the isolates tested over the last 4 years infected LR24. And virulence for LR26 has dropped to 50% in 2021. Virulence for LR28 has continued to increase again in 2021. And virulence for LR37 and Crusoe have remained at 100% for the last 4 years. So these are now going to be, that pop up the resistant genes, the virulence frequencies for the Thatcher line resistant genes, which we introduced to our differential set in 2016. So none of the isolates tested over the last 5 years infected LR2A, or over the last 4 years infected LR2B. Virulence for LR2C has remained stable over the last 3 years, and virulence for LR3BG has dropped again in 2021. Virulence for LR3KA dropped to 50% in 2021, and virulence for LR10 has increased again to 100%. And all isolates tested successfully infected LR13, LR14A, LR15, and LR17 for the last 4 years, and LR16 for the last 3 years. And virulence for LR23 has increased in 2021, whereas beforehand we were seeing it decrease over the last 4 years. Now moving on to pathotype frequency for 2021. This graph shows the percentage of them 10 isolates tested for each each of the pathotypes, and denotes one of the pathotypes identified. So the most dominant pathotype had a virulence, had a frequency of 40%, and here is its pathotype. It was also present in 2020, this pathotype. And the second most dominant pathotype had a frequency of 20% and had this pathotype. This pathotype was present for the last 4 years and was also the most dominant pathotype in 2019. There also was the only pathotype to have any correlation between location, with all of the samples with this one coming in from either Berkshire and Oxfordshire, but obviously with quite a low sample size, it's difficult to draw any conclusions. And then only one sample had one of these pathotypes, with one of them being the only new pathotype for 2021 with its pathotype, and it came in from Cambridgeshire on the variety relay. So here are the 2021 isolates selected for this year's adult plant trials. Again, will be tested on recommended list and candidate varieties, which will be inoculated this summer. So 21002 was selected because it came in on the variety KWS Kerin, which has an RL rating of a 7, and its pathotype carried virulence for LR2C. Isolate 21003 was selected because it was a new pathotype for 2021 and also carried virulence for LR28. 21004 was selected because it's the second most dominant pathotype in 2021. And 21006 was selected because it came in on the variety KWS Cranium with an RL rating of a 4, and its pathotype carried virulence for LR20 and 28 and was the only sample to come in from Lincolnshire. And 21008 was selected because it was the new pathotype. I just said 21003 is the new pathotype. That's actually the most dominant pathotype. So the results for these will be presented in the UKCPVS report in September. Just to take a little look at the wheat brown rust recommended list ratings for 2022-23. So most of the varieties, few varieties had an RL rating of an 8, and then none of the varieties had an RL rating of a 9, which is reflective of LG Astronomer dropping from a 9 to an 8. And then most of the varieties had an RL rating of a 7, 6, and 5. One variety had an RL rating of a 4, KWS Cranium, and then 2 varieties had an RL rating of a 3, which is Crusoe and KWS Guion. So here's the wheat brown rust summary for 2021. There's moderate to low disease pressure arriving late in the season, which we think was possibly linked to the weather. Seedling virulence frequencies increased for the first time. In 4 years for LR20 and LR23, and continue to increase for LR28 over the last 4 years. Seedling virulence frequencies have decreased significantly for LR26, and isolates tested remained avirulent to LR24 for the last 4 years. And a prevalent pathotype was detected for 2021. So moving on now to the wheat brown rust, to the wheat powdery mildew results for 2021. Ooh. A little bit of wheat mildew, a background on wheat mildew. It's been monitored since the beginning of the survey. Resistance to mildew is based on PM genes and other unnamed genes. And historically, there were issues with swift breakdowns in resistance as the PM genes were overcome, but now it's a more stable situation. Here are the 2021 wheat mildew samples received. We received 10 samples from 6 counties and 3 varieties, and there were 6 unknown varieties. Unfortunately, 6 of those samples were unsuccessful out of the 10 received, and the 3 varieties that were known were all RL varieties. So we can see most of the samples came in from Suffolk, followed by Cornwall and Yorkshire, and 8 single partial isolates tested were tested from the 4 successful samples. So here is the current wheat mildew differential set and its resistant genes, which all the isolates were tested on, and here is a picture of one of our detached leaf tests. So from those 8 single partial isolates from those 4 successful samples which were isolated, they were tested on that wheat mildew differential set to determine their pathotypes. And again, this graph shows the percentage of those isolates tested over the 5 years which carry virulence for the resistant genes which will pop up. You may notice the year 2019 and 2020 are missing, and this is because unfortunately that year the few samples that were received were. So for virulence for PM3B and PM17 increased to 100% in 2021, with virulences before that being quite low. Virulence will still continue to increase again in 2021 to 100%. Virulence for MLSH has previously from 2015 to 2018 been at 0% and went up to 50% in 2021. And virulence for Armada has previously been for the last 4 years at 60% plus and is now at 0% in 2021. Virulence for PM3D has continued to increase in 2021, and virulence for Crusoe has remained to 2016 and 2017 levels. Virulence for PM2 and MLD have both dropped from previously being at 100% in 2018, and virulence for Mercia has been at 100%. Was at 100% in 2017 and seems to be dropping back down again in 2021. So now just to look at the pathotypes. So there was not one single dominant pathotype, there was 8 pathotypes from 8 isolates tested, and all of those pathotypes, 8 pathotypes, were novel from 2021. And we believe this is mainly due to those increases in virulence for PM3B, PM17 and MLSH, and virulence dropping for PM4B. Which if I just take you back to that graph bar chart that we were looking at earlier, it's these 4 genes, resistant genes, and we can see that in 2021, for the red bars, we can see how much they've changed, or in Armada where it's completely dropped off, compared to the previous 2015 to 2018. So just to summarise, some resistant genes saw significant seedling virulence frequency changes compared to previous years. Virulence frequencies increased to 100% for PM3B, 17 and STIG, and to 50% for MLSH, and dropped to 0% for PM4B. 8 new pathotypes were identified for 2021 with no single dominant pathotype. And although there were some changes in virulence frequencies seen, this was not linked to any reports in the field. So I believe now we'll hand you back to Charlotte.
Thank you. Okay, so just a few more slides to get through. So I just want to talk about the sampling for the coming season. So have we seen any rust yet? So we have received a few early season samples from KWS Zayat and Skyfall from Kent, so these are yellow rust samples. And we've also received a few early season samples from barley mildew. So as has been mentioned throughout this presentation, that the survey really relies on people to take time and send us in the samples, and we really appreciate the time and effort that everybody goes to. So please, if you do see any wheat yellow rust, wheat brown rust, or wheat and barley mildew, please do send us in some samples. Yeah, and these are welcome from any RL or RL candidate variety, preferably, well, definitely from an inoculated field, and preferably from untreated trials. We've had historical lows for mildew samples, so if you do see those this season, we would appreciate you sending those in. This is our sampling instruction sheet and form. You can find this on the website. There's a link at the end of the presentation, and please send that in with each of the samples. You can post it free post to UKCPVS. Please only use paper envelopes for all the pathogens, because if you use polythene bags, the samples won't survive and we won't be able to recover anything. And if you think something is really important or really interesting and you want to send it with a stamp, please use the address at the bottom of the slide to our Park Farm site. Yeah, and so further information can be found on these websites, which are listed in the handout. So please do check those resources out. Whilst you're here, I just wanted to draw your attention to a conference that we're hosting later in the year. It's been postponed from April. So it's now being held on Wednesday the 31st of August to Friday the 2nd of September. And this is at Clare College in Cambridge, and it's the 16th International Cereal Rust and Powdery Mildew Conference. So please do check out the website and come and join us in Cambridge in August, September. So finally, I'd just like to acknowledge the funders that fund this work. Thank all of you guys for sending samples to us, and also the team that are based at NIAB, Jane, Judith, Helen, Anne, Megan and Edda, and also Lucy, sorry, my predecessor, and all the field trials team. Do you have any questions? Hopefully some of them have been coming through in the chat function, but if you think of anything later on, please feel free to email myself or Amelia. Thank you very much for your attention.
Well, thank you very much to the UKCPVS team there of Charlotte, Amelia and Sarah for getting through all that data that's you know, formed the basis of the survey for the last year, and some very interesting points there. We do have an opportunity to look at questions briefly before our break. So I'm just having a quick look on the questions pane here. I don't know if Catherine's been having a look as well.
Yeah, I can read out the questions we've had through, if that helps.
Yeah, I've spotted one here about the cutoff of 2.4 selected for deciding on the isolate virulence or avirulence. So if the team there could think about that one and maybe come back on it. So why has the 2.4 rating been selected for deciding on the isolate virulence, avirulence?
I can't actually see the questions. Sorry.
This was posted, I think, by Kostya Konyuka.
If you open the questions into a different window, then you can see all the questions.
Okay.
There's relatively few in my window, but that's the first one I can see that's directly concerned with the ratings.
I still can't see anything, I'm sorry. Can you just repeat it? Sorry.
Okay, so it's to do with the yellow rust resistance scoring. I wonder how the cutoff of 2.4 has been selected for deciding on the isolate virulence, avirulence from Kostya.
Amelia, should I hand that over to you?
Hi. So yeah, we have a cut-off point for 2.4 because we actually have a 0 to 4 scale, and we have sort of like a set guideline of what we would score as a 0, what we would score as a 1, and a 2 is basically an incompatible reaction because it has very few pustules, very sort of like low sporulation, and then our 3 and 4 used to actually be our susceptible sort of category. Type 3 is lots of pustules, lots of sporulation, and we found in our seedling tests we were deciding that actually things that had a 2.7, so basically the majority of the leaves that we score are a 3, but with a couple of 2s in there, that's now susceptible. So that was where our cut-off point for the 2.7 come from. But the lower end of a cut-off point with the 2.4, that we just wanted to highlight basically that there is a borderline reaction between varieties. So it could be that although we don't class it in the pathotype as being virulent if it's got a 2.4 reaction, it's just useful for us to go back and have a look at those results and say, well, actually, yeah, it was a bit with borderline in our test. So maybe under different conditions, different, you know, just different lighting or different temperature or something, maybe that 2.4 in another world could have gone either way. I hope that answers that question.
Thank you very much, Amelia. That's very helpful. There was just a query from John Miles, who I think actually possibly has left the meeting, on nothing from KWS X days in 2021. Question.
What do you mean nothing from KWS X days?
That one's been answered. I think they, during the presentation, he asked it, and then he says later it's been answered. So you answered the question within the presentation, so that one's okay. But we do have another one. There are increasing number of RL wheat varieties with low powdery mildew resistance ratings. Is this related to the increasing frequency of virulence in isolate against many resistance genes?
I guess I could have a stab at that. I guess it's quite difficult to say because we don't get that many isolates, but the maybe Amelia has a better memory. Sorry, I'm just new to this project and can't remember all the details off the top of my head.
Yeah, sorry, I'm slightly missed the question there. So
there are increasing number of RL wheat varieties with low powdery mildew resistance ratings. Is this related to the increasing frequency of virulence in isolates against many resistance genes?
So I don't know if it's actually related to our results, because we just don't get that many samples coming in. We had quite a few this year, but previous years we've not had anything to test. So I think we would be able to answer that question better if we get some more samples in, especially for wheat powdery mildew, because yet we do see quite a few varieties. They get it in the spring, it likes the hue, you know, the sort of like the cooler and the wet conditions of spring. So we do see quite a bit of powdery mildew out there, and with some more samples we'll be able to do a lot more work for that.
Okay, thank you very much, Amelia. There are now a number of questions appearing. I think in view of the time, because we're going to take a 10-minute break before Diane's presentation, and in fact it's now 11:42. So is everyone happy if we can have a break now? And of course we do have a Q&A session at the end where we can hold back any of the questions that haven't been answered at this point and reconsider them in the final conclusion debate. So unless anyone disagrees, I think we perhaps ought to have a break at this point, and we can reconvene in about 10 minutes. Okay. Thank you very much to the team for all the information. Hello.
Hey, John.
Welcome back, everybody. Hope you had a good break. It's now my pleasant duty to introduce Diane Saunders, who's a project leader at the John Innes Centre. Diane, as you've already heard, has played an important part in developing field pathogenomics approaches for characterising rusts. Today she's actually going to talk about a new foe, which is actually wheat stem rust. So over to Diane.
Thank you very much, John. I hope you're all feeling refreshed after your break. So today I just wanted to highlight a disease of potential concern for re-emergence here in the UK, which is wheat stem rust. And so wheat stem rust is a very devastating disease, as you can see here. It occurs in most major wheat growing areas around the globe. And here in the UK, it used to be quite a frequent disease. We'd see frequent epidemics. However, the last epidemic that occurred here was back in 1955, where we saw average yield losses reaching around 50%. And then the disease seemed to largely disappear. However, this situation then changed in 2013, when there was one infected wheat plant infected by wheat stem rust that's shown here that was identified down in Suffolk. And so this is just one infected plant, not a big problem for agriculture, but it is, of course, rather significant because it was the first time that we'd recorded wheat stem rust in the UK for over 60 years. And also in the same year and in subsequent years, many of our colleagues across Western Europe recorded wheat stem rust in their fields. And in many of these locations, this was the first time that wheat stem rust had been recorded in these Western European countries for a number of decades. And then in 2020, our colleagues across the water in Ireland also identified wheat stem rust at 5 different trial sites. And luckily, this was just in untreated trials. But again, rather significant because this was the first time that wheat stem rust had been seen in Ireland since the 1960s. And then just last summer, we were also notified to a number of wheat stem rust incidences again down in Suffolk. And in this case, we've gone from a position in 2013 where just one infected plant was recorded to having handfuls of plants being infected at a small number of locations. And so then seeing this increase in potential sporadic incidences of wheat stem rust, the question then is this likely to continue into the future? And so to address this question, we worked with a climate modeller, Dan Bever at Exeter University, and he looked at the environmental conditions that are required to support wheat stem rust establishment. And he found that over the past 25 years, those conditions that we need in the environment to support wheat stem rust infection have been increasing and becoming more and more conducive to supporting infection. And of course, this is then reflected in the fact that we're seeing more sporadic outbreaks of wheat stem rust across Western Europe. And so the question that then comes to mind, seeing this increase in sporadic outbreaks, is why did this disease disappear in the first place? And so why haven't we seen any epidemics here in the UK since 1955? And I think there are a number of different factors that have protected us against wheat stem rust outbreaks here in the UK. One of these, of course, is the important contribution of plant breeding. Breeding firstly for early maturity. So wheat stem rust is a warm temperature disease. It tends to occur in the summer months, and breeding for early maturity means that the plants then senesce a bit earlier. And so that green tissue isn't in the field for the pathogen to infect in the summer months. Also, of course, introducing resistance is very important. Then we have the removal of the alternate host of the pathogen, this Barley, this hedgerow species, which I'll return to and explain a bit more about why it's important later on. But it's very important in supporting the sexual reproduction of the pathogen, and so removing Barley prevents the pathogen from having sex here in the UK. And that then reduces the number of races we see of the pathogen. Also, it's very important in facilitating overwintering of the pathogen in cooler climates, such as the one that we have here in the UK. And so it's a very important in the role of the life cycle of the pathogen. Also, of course, we now have modern fungicides, and they're very effective in controlling wheat stem rust. And so when we think about all of these factors that have protected us against wheat stem rust outbreaks, it really took an entire community to prevent out any epidemics in the past 60 years, right through from the plant breeders, the ag chem companies, the plant pathologists, through to the policy makers. However, looking at the number of sporadic outbreaks that we've seen across Western Europe in the recent years, the question that then comes to mind is how many of these still remain effective today. And so if we firstly consider plant breeding, we decided to then look at the amount of resistance firstly that was present within UK wheat crops. And so to do this, we worked very closely with Jane Thomas at NIAB, and she took around 40 different wheat varieties, many of the varieties from the UK recommended list. And then she infected these varieties at seedling stage with a UK isolate of stem rust, the one that was collected back in 2013. And she found from this analysis that over 80% of those varieties that she tested were susceptible to that wheat stem rust isolate at the seedling stage. And so we also wanted to test these varieties at the adult plant stage, and to do that, we here in the UK, of course, we don't get very much infection with wheat stem rust, so planting them out in the field wouldn't really be an option. And so to do that, we then worked with a collaborator, Willem Boshoff, at the University of the Free State in South Africa, and he planted out nearly 60 different UK varieties and then scored them in the field after they'd become naturally infected by the isolates of stem rust that were present in his fields in South Africa. And what he found from a number of years of doing these field trials was that all of the varieties that he tested were very susceptible in these field trials, at least to the isolates that were present in South Africa. And so from this analysis, what we can conclude is actually there is very little resistance in our UK wheat varieties to the wheat stem rust isolates that we were testing in these experiments. And so then the next question is regarding that early maturity and whether the early maturity, at least of our wheat crops, will protect us if wheat stem rust became more frequent. However, climate instability is now starting to threaten that early maturity in a number of different ways. And here is just one of the ways that I wanted to illustrate today. And so if we go back in time to the autumn of 2019, many of you might remember that was a very wet autumn, and we're seeing an increase in the number of these kind of very wet autumns here in the UK. And you can see this farmer's field here. And of course, this was waterlogged. And so in the autumn of 2019, it was very difficult to get out there and plant your winter wheat crop. And so this then created a bit of a dilemma for farmers because they couldn't get into their fields in the autumn. And so in the 2019-2020 season, of course, they were threatened by this large deluge of rain that occurred in the autumn, and they had to make a decision as to how to deal with this. And what some farmers then decided to do was to switch and wait until the rains had cleared and to plant spring wheat instead. And of course, if we look at the data, here is data that's taken from Ireland that was readily available. You can see that many people decided to do this. If we look at the area that was allocated to winter wheat, this decreased in the 2019-2020 season by around 40%. The amount of spring wheat was around 200% as farmers decided to plant more spring wheat once the rains had cleared. But unfortunately, that then presents a potential issue because spring wheat matures a bit later than winter wheat. And so that early maturity that we have with our winter wheat crops is being eroded if we then plant spring wheat, as the spring wheat is nice and green out there in the fields much longer and into those summer months where wheat stem rust could potentially take hold. And so by switching our planting strategy and planting more spring wheat in light of these kind of really awful autumns that we're seeing with more rain in the autumn period, this could actually then have a knock-on impact by enhancing the incidence of warm temperature diseases that then could take hold as we're eroding that early maturity. Okay, so then moving on to the alternate host, this barberry, this hedgerow species. This barberry plant is very important in the life cycle of our wheat stem rust pathogen. And just to explain its importance, I wanted to just touch on the life cycle of the wheat stem rust pathogen, which is very, very complicated. It has 5 different spore stages and 2 different hosts. And if we draw our attention to the right-hand side of this diagram, here is where the pathogen is cycling on wheat. During the summer months, it will produce vast quantities of asexual uridinospores to go through the summer months, moving on the wind from one susceptible wheat plant to the next. But then if we draw our attention to the center part of this diagram, once the summer is over, the pathogen really switches to a different type of spore type. And so it will then produce these hardy overwintering teleospores, because wheat stem rust can't survive through our cold winter months in its asexual form. And this is very much unlike yellow rust, which is capable of surviving in its asexual form through the winter months. Stem rust can't do this. It moves and starts to create hardy overwintering black teleospores on like volunteer grasses or indeed on any wheat stubble in the field. It will then wake up again after it's survived through that cold winter, and in the springtime these teleospores will then start to create basidiospores, and those basidiospores are what then go on to infect the alternate host of this pathogen, which includes many species in the Berberis genus, including common barberry known as Berberis vulgaris, which is very susceptible to wheat stem rust. And once it's infected its alternate host, this is the point where the pathogen then completes sexual reproduction. And so this means that the pathogen then creates new genetic variants of itself that might have new characteristics. And those new genetic variants then are produced in a different type of spore, these asiospores, that can't reinfect the barberry bush. They have to go on and find a serial host for infection. And so the barberry bush has 2 real roles. It helps the pathogen to complete sexual reproduction, but it can also act as a very important source of inoculum for then going on and infecting cereal crops. So if we look into the future and the potential for us, unfortunately, if we did have a wheat stem rust epidemic here in the UK, there would be really 2 different sources that this could come from. And so, for instance, we could have the asexual uridinospores starting an infection here in the UK. However, the quantity that are created and needed for initiating a really large-scale epidemic, well, these would likely actually originate in southern Europe, where it is much warmer. The pathogen can get going much earlier in the season, and then you could imagine they could potentially blow into the UK later in our summer and start an infection. But hopefully they would arrive after our plants have started to senesce. But the pathogen also has the option to create in-country inoculum in the form of asiospores, and these can be created locally on these barberry bushes, and they can form much earlier within the growing season. And indeed here within the UK, some work that was done by Elizabeth Orton in the lab has shown that in the UK, the pathogen is capable of switching through its whole life cycle. So going from the barberry bush, where it has caused an infection as shown here, and then being identified in a neighbouring cereal crop. In this case, we have infections on barley that were identified right next door to this barberry bush, and wheat stem rust can also infect barley. And so from this work, we can say that the pathogen is able to complete its whole life cycle here in the UK, and that barberry could act as an important source of inoculum. But the fact that barberry is important in the life cycle of wheat stem rust is not a new fact. It's been known for a very, very long time. And indeed, it led to a lot of legislation and policy to enforce its removal and exclude it from locations around the globe, including many countries in Europe, also in America and Canada. And I think one of the campaigns that has been best documented is the one that occurred in America. And these are just some of the posters that accompanied that campaign, which ran from the 1910s into the 1980s, where they enforced removal of barberry bushes. And indeed, millions of bushes were removed in this period in North America in the wheat growing areas. And this was very successful. It reduced the number of races that they were seeing, so the number of different genetic variants, from around 17 races at the beginning of the period in the 1910s to around 6 or 7 per year by the 1980s when the campaign stopped. It also importantly removed that local source of inoculum, so the scale of epidemics was much smaller because you didn't have that local source of inoculum. And indeed, the fact that it's very effective as a control measure against wheat stem rust means that policy and legislation still today continues to limit barberry plantings, particularly in America and Canada, but also in some countries in Europe as a very effective control measure for wheat stem rust. And recently in the lab, we also revisited the history of barberry here in the UK, and we showed that it was actually introduced into the UK in the Middle Ages. And as long ago as around 3 centuries ago, our farmers started to rip it out. And we didn't have any legislation or policy to enforce removal here in the UK because our farmers were just so proactive. They noticed that if they had these bushes on their land and they looked infected, then their crops got infected too. And this was before scientists had really documented this link between these two different hosts. And so our farmers noticed this link and they just pulled these bushes out actively. However, of course, now with wheat stem rust being a very distant memory here in the UK, the number of these plants is starting to increase. And if we look just at Berberis vulgaris, common barberry, which is very susceptible to wheat stem rust, the number of these plants has increased, and you can see the distribution. And they're found really all over the UK, especially in our wheat growing areas. We can readily identify these bushes in those regions. And also there are efforts to actively plant Berberis vulgaris, common barberry, which is very susceptible to wheat stem rust here in the UK, because it's also currently the only known food source for an endangered moth species, this Barbary carpet moth. And so there are small efforts for local replanting of this hedgerow species to increase habitat for this endangered moth. And so now we've got ourselves into a situation where we have a lot of this common barberry in the UK. And so the question we've been trying to address is, how do we manage the risk of all these common barberry plants if we do start to see wheat stem rust continuing to cause these kind of sporadic infections going forward? And so to address this, this work was then conducted by a very talented PhD student in my lab, Vanessa, who decided to address this by looking at how much inoculum, so how many spores are created on a barberry bush and how far they might travel. Because really what we were trying to do was to work out how far they could potentially travel to ultimately inform policy around planting strategies, to thereby kind of limit them planting away from cereal crops at a safe distance. And so to address this, we had to look at these aciospores and how they're released from these infection structures. And these aciospores that are created in these tube-like structures that form on the underside of barberry leaves in these kind of clusters, they are released in vast quantities from these infection sites when you add water, such as is shown in this movie here. You can see all these spores being spurted out of these tube-like structures. And so we wanted to work out how many spores are really being released, not just from one infection site, but from a whole single bush. And to address this, we generated a huge amount of data, which I shan't go through today. I just wanted to highlight a few of the key elements of those data sets. So Vanessa looked at these single infection sites and these tube-like structures, and she looked at how many spores were created in a single infection site. And so by looking at the structure of these tubes, she could measure their length. She could look at how many layers of spores you would have within a single tube-like structure, how many of these tube-like structures on average were present in a single infection site. And then she could calculate the likely amount of spores that you would have in a single infection. And she found that this was around 250,000 spores that were present within a single infection site. She also went to the field and she collected lots of leaves that were infected by stem rust and then classified them into different levels of infection. And then she looked at various bushes out there in the field, and she also looked through the literature to look at, on average, how many of these leaves would likely be infected in a single bush, an average size single bush. And so from this analysis, she could then kind of have an estimate on the number of spores that would be released from a single infected bush. And then she could put that information into a very simple model and then work out how far those spores could travel based on current weather conditions. And so she put that information into a web interface that she developed, which is a point and click web interface. And you can now go to this web interface, you can put in your current location, roughly how infected the bush is, and it will then predict how far the spores will travel using current weather data from that location. And so by creating this model, she could then look at the density of spore dispersal projections, and then hopefully this information can help to guide policy around safe planting distances for barberry hedges near cereal crops, and also to identify any hedgerows that might pose a high risk to cereal crops so they can be then in the future carefully monitored. So as we look into the future and our preparedness for the potential for wheat stem rust re-emergence in Western Europe, and in particular here in the UK, we now know that there is limited resistance within our UK wheat varieties that we're growing out there in the field. And we need to be a bit mindful about any changes to our wheat planting strategies, such as moving from winter wheat to spring wheat, and how that could impact incidences of warm temperature diseases such as wheat stem rust. We know that barberry is present all around the UK, and we've now developed this really nice ACO spore dispersal model, which allows us to assess the risk of any new plantings and make sure that they're planted far enough away from cereal crops, and hopefully to inform policy as we go forward. And I haven't talked much about fungicides, because fungicides still remain very effective in controlling wheat stem rust. There are no incidences on a global scale of field failure from fungicides that are used currently to control wheat stem rust, but of course they are just one element in a disease management strategy, but they are indeed very effective. And so as we look into the future, of course, controlling wheat stem rust and ensuring that we maintain the UK as being absent from wheat stem rust epidemics, of course, this will take an entire community, right through from the plant breeders to the ag chem companies to the plant pathologists and the policy makers. And so I'd just like to leave things there by acknowledging, of course, all of the people in my lab who have played a role in the various work that I talked about today, all of our local collaborators and our international collaborators, and all of our funders. And thank you very much for your attention today.
Thank you very much, Diane, for that nice piece of horizon scanning and risk assessment. I think this is the kind of preemptive study that is now, very important going forward. I think in view of the time, we haven't got much time for lots of questions at this point, but there is one here I spotted, which was about the virulence genes present in the race of stem rust used for the seedling tests. I don't know if there is an answer for that. I think that work was probably done by Jane Thomas.
Yeah, so we do know what genes were present, and all of that information is in the related publication. It's a race of the stem rust that is prevalent across the whole of Western Europe. So we do have information about those particular genes.
Okay, that came, I forgot to say that was from David Furhelm.
Yeah, and if you want to get in touch and talk about it more, then just drop me an email. Happy to discuss further.
Okay, thanks very much. I think we ought to move on. So it's now my duty to introduce the next speaker, who is actually Rachel Goddard from Lima Grain. Highly relevant topic in view of everything else we've heard. Challenges for breeding rust resistance post-warrior. So over to you, Rachel.
Hi, thank you for the introduction. Are you able to see my screen?
Yes, I think we're here.
Great. Okay, that's perfect. So I really wanted to start this talk with this slide, which has the yellow rust timeline pre-warrior. So the major theme of this slide is resistance breakdown. So this is essentially a major shift in resistance, so from resistant to susceptible over a short period of time. So if we use Solstice as an example, it had a recommended list rating of 9 in 2008, and then this dropped to a 4 in the subsequent year. So this arrow is essentially a timeline of the major resistance breakdowns over the past 50 years in the UK. So beginning with Rothwell-Perdix in 1966, which we've already heard about, and then ending in 2009 with Oakley Solstice. And we can see from the dates on the timeline that these breakdowns occur kind of once every 5 to 10 years. And these breakdowns are not completely random, and they're often in direct response to the wheat varieties which are grown at the time. So if we look at this schematic here, so pre-warrior, the UK rust population was clonal, and we can see that there would be one dominant race at a particularly given time. In response to a wheat variety being grown over a larger area, new variants would often arise, which is kind of represented by this yellow circle. And then over time, this new variant would be the new dominant race. So this is essentially stepwise changes in virulence, which occur every few years. If we move to 2011, this was when the warrior race was first identified in the UK and Europe. And this was essentially a complete population change in yellow rust. So warrior was highly unusual for a number of ways. So firstly, in that this race was identified in multiple countries throughout Europe in the same year. Secondly, warrior broke a number of effective resistances in European winter wheat germplasm. So many of these resistances were no longer effective. And also, compared to the previous clonal yellow rust populations in the UK, warrior appears to have been derived from a sexually recombining population outside of Europe. So if we contrast this from the simple model which I showed you on the previous slide, so a single new variant in an otherwise clonal population, we then have warrior, which is essentially a population of variants that can readily attack. Oh, hello?
Okay.
Okay. So since 2011, yellow rust has remained very complex in the UK. I'm sorry, I can't see my screen anymore. Yeah, there we go. Yeah, so as has been mentioned, we have a new naming system to reflect this complexity, and then the pathogenomics work has identified that there are several genetic groups present in the UK. And whilst there has been some fluctuation over the past few years, we can see that the red genetic group is dominant, and that this is very diverse with over 50 red pathotypes. We can see this diversity in the field. So this is work which was undertaken by Chris Judge at NIAB, and he identified that different pathotypes can be found within the same field, and also that the frequency of pathotypes can change throughout the growing season. So for example, pathotypes which are present at the beginning of the growing season can be completely different to pathotypes later in the growing season, even in the same area of the field. So this means that it's quite difficult to predict the rust population, how it might change, because the situation is very fluid. So this slide just shows kind of the optimal yellow rust conditions pre-warrior. So yellow rust is a biotroph, which means that it needs green leaf tissue in order to survive. And the fungus prefers milder winters, which mean that it can overwinter on volunteers or autumn sown crops. And cold winters and frosts kind of really slow development, particularly if it kills off the lower leaves. In spring, yellow rust can tolerate warmer temperatures, but it really needs cooler evenings with overnight dew in order to develop. But since the warrior race has become prevalent, rust is now much more adaptable and able to withstand greater changes in the environment. So for example, rusts are normally slowed by temperatures of over 20 degrees C, but the warrior race is able to tolerate a greater range of temperatures. So we can now see rust in countries where it was previously thought to be too warm for rust development, such as Italy and Spain. Warrior also has a shorter time from infection to sporulation, and this means that there's a potential for the disease cycle to repeat several times during the season. And warrior is also much more aggressive, and it produces more spores than the old yellow rust. Warrior also looks quite different to the old yellow rust. So for example, now we can often see these black telia on the leaves instead of the yellow pustules in stripes. And this is also an indication that warrior has evolved from a sexually recombining population. So as I've described, yellow rust post-warrior is very different to the old yellow rust, and we can essentially consider it to be a new disease. And this has really changed the way that we have been able to breed for resistance. I wanted to just have a couple of slides to show how warrior actually has changed resistance breeding, and I thought it would be good to begin with Clare. So following the breakdown of Brigadier in 1997, the variety Clare, which was high yielding and also had good all-round disease resistance, was used extensively in breeding, particularly because it had a single adult plant resistance gene, which we call YR Clare, and this provides significant resistance to yellow rust. And this graph here just shows the disease susceptibility of Clare in the Limmergrain disease nurseries over a period of 20 years. And the red line shows the maximum score for Clare, and then the blue bars show the mean score. And we can see that for around 10 years, this YR Clare gene was providing significant resistance in the Clare variety. However, after the appearance of the warrior race in 2011, we can see a marked increase in susceptibility to yellow rust in the variety Clare in 2012. And then if we look at the more recent years, so 2019, 2020, we can see that Clare has a disease score of around 60%, so it's very susceptible to yellow rust. So the warrior race has essentially broken this YR Clare resistance, which provided significant resistance in the UK wheat population. And we now think that Clare is a susceptibility factor. And I can actually contrast this with Brigadier. So as I mentioned, Brigadier resistance broke down pre-warrior. And again, the graph shows the disease score of Brigadier in the Limmergrain disease nurseries, and we can see that this variety was susceptible. However, if we look in more recent years, so from 2017 onwards, and this is when there's less fluctuation in the different genetic races which are present in the UK, we can see that Brigadier is actually now very resistant to yellow rust. And this means that Brigadier has YR genes which were defeated by old clonal YR races, but they're now providing resistance against the warrior population. So this is just an example of two complete shifts in resistance breeding in the UK. So I just wanted to talk now about how we're actually resistance breeding post-warrior. And there are really a number of important points which you have to factor into your resistance breeding strategy. So firstly, we've heard about the diversity in the UK rust population. And this means that it's really difficult to predict how the population will change within the next 5 years or even the next 10 years. There might potentially be new exotic incursions, and climate change may also have an effect. It's also difficult to predict which host resistances are durable. So durability is essentially resistance which remains effective during prolonged and widespread use in an environment which is favourable to disease. So this is obviously something which we're striving towards. And we know that single genes which are deployed on their own are unlikely to be durable. So we know that we're looking at combinations of genes together. However, it's difficult to predict which combinations are likely to be durable. So there's still some uncertainty there. Thirdly, I think it's also important to recognise that a traditional wheat breeding cycle takes around 10 years. So this is from initial cross, nursery selection through to yield trials, and then national list and recommended list trials. And this means that crosses which were made 4 or 5 years ago may be less resistant by the time that they reach recommended list trials. And this is just due to the diversity and the change in the rust population. So really, trying to breed for resistance to rust post-warrior means that we have to use all of the data which is available to us and try and make the best and the most informed choices that we can. So regarding the pathogen population, we can gain a greater understanding using the UKCPVS data. So as plant breeders, we work on wheat host genetics and not the pathogen. And this means that the work of the UKCPVS allows us to understand if there are any race changes. So it's really an early warning system. And it's also providing us with validation of new races. Also the identification of new pathotypes and which virulences are present and at which frequency. And we also make sure that we use the official rust races, which are supplied by NIAB, to inoculate our disease nurseries. And this means that we are not relying purely on natural infection, which we know can be slightly different depending on the location. And this means that we're not selecting breeding lines that might be susceptible to new races. We also take a lot of time to understand the resistance which is already present in elite UK wheats. So AHDB now provides a yellow rust watch list, and this is essentially giving an indication of which varieties might be more vulnerable to a ratings fall in the coming season. This is done by taking the recommended list trial data for a variety and then comparing it to other varieties. And then each variety is grouped into 3 categories. So those with high resistance, which is varieties which are highly resistant in all trials. And these are shown in the light blue colour, those with medium resistance, and then those with low resistance. So these are varieties which are highly susceptible in at least some of the trials. And this graph is essentially just showing the AHDB watch list rating for the recommended list varieties versus the number of effective wire genes that each variety has. So from 0 to 4 genes. And then we have the number of lines in each category on the y-axis. And I think it's quite clear that varieties which have 3 or 4 genes have greater resistance. And we know that there are at least 8 different effective resistance genes for yellow rust in recommended list varieties currently. However, these are found in different frequencies, so some are more common and some are more rare. And we also know that not all R gene combinations are effective against warrior. And again, it's just important to note that crossing to entry takes 7 to 10 years. So varieties which are now looking to have lower resistance is due to the changes in the pathogen population. We also take care to monitor these resistances in our disease nurseries. And this is because, as has been previously mentioned, the pathogen population is very complex and it changes yearly. So this graph shows different yellow rust gene combinations. So each point on the graph is a different combination in our different disease nursery locations, with the disease score on the y-axis, so 10 being the most susceptible. And we can group these combinations into 3 rough categories. So green, which is combinations which are providing high levels of resistance, those in orange, which is moderate resistance, and those in red, which are susceptible. And this means that by monitoring these resistances year by year, we can see which gene combinations are perhaps shifting from the green category into the orange category. So they're potentially losing efficacy. And this also means that we know that we can't rely on a single gene, and we also don't want to rely on single gene combinations to provide resistance. So by monitoring multiple resistances, this means that we can try and maintain some diversity within our breeding program. We could also try and introduce new resistances into the breeding program via pre-breeding. So it's been mentioned before, but breeders have been successful at increasing resistance. So if we look at the graph in the top right of this slide, this is essentially just looking at YR susceptibility versus the number of R gene clusters. And this is a comparison between elite varieties, which are the green circles, and then plant genetic resources, which are in yellow. And we can see that in the right-hand side of the graph that there are a number of elite varieties which have a greater number of R gene clusters which have been introduced, and they're resistant to yellow rust. However, the elite wheat gene pool in the UK is quite narrow. So if you cross elite varieties with other elite varieties, then this really produces a limited number of YR combinations. So we might want to try and introduce resistances from elsewhere. This pie chart shows the identified yellow rust resistance genes and essentially their sources. So the big blue section of the pie chart are resistances which are native to bread wheat, and we can see that this is kind of 75% of the pie chart. So maybe we might want to tap into the resistance from this smaller section of the pie. And there are a number of resistance sources which we can try and exploit to expand the resistance gene pool in elite wheats. So wheat has three gene pools. So we have the primary, which is elite wheats, land races, and genetic stocks. And then we have the secondary and tertiary gene pools, which are essentially made up of wheat progenitors and crop wild relatives. So we can try and bring in some more diversity and some new resistances from the secondary and tertiary gene pools in order to try and combat the changes which we see in the warrior race. And then assessing elite sources of resistance is kind of part of what we're doing with work with the Designing Future Wheat, or DFW program. So whilst there's a wealth of diversity in terms of resistance in these secondary and tertiary gene pools, we have to consider that these are essentially, they're not really a magic wand, or they don't solve all of our problems. And this is because non-native sources are often very tall, so they're prone to lodging in the field. They often have lower yield. We can see from the diagram that these diverse sources often look very different to elite wheat, and it's often difficult to even cross these varieties to elite wheat, so we often experience issues with sterility. And then because of all these reasons, the introduction of non-native sources into breeding programs takes 2 to 3 times as long as elite sources. So this means that we really have to be selective with which resistances we think will be useful, particularly due to the changes in the pathogen population and how quick that they're occurring. And finally, once we've introduced all of these resistances into our breeding programs, then we want to try and protect them so that they're effective for as long as possible. And we can do this by stacking genes together, as this provides more durability. So we can see from the graph, so this is just looking at the different number of genes which are stacked together, so 1 to 4, and then the disease score. And we can see that varieties which have 3 or 4 genes stacked together are again more resistant. However, in order to create lines which have effective gene stacks of, for example, 3 or 4 genes, this really requires thousands and thousands of lines to be able to create the right combinations. And we also need to consider the effect on yield that this has. So for example, if you're stacking multiple genes together, this can also have a fitness cost on the plant. And we also need to remember that yellow rust resistance is only a single trait. So if we're looking at recommended list diseases which we need to consider, we've got brown rust, we've got septoria, mildew and fusarium. And this also isn't taking into account different quality traits and obviously yield, which is incredibly important. So it's really trying to balance disease resistance and the other important traits which we need in a variety. And to stack genes together really requires genetic markers. So if we look at a resistant variety in the field, we don't know if this is due to one gene or a stack of genes in combination. And this means that if you are purely selecting two resistant varieties and crossing them together, you don't know if you're creating new combinations or the same combinations. So you really need genetic markers to pick specific resistances and bring these together. So we have to kind of take into account all of these reasons, and we also have to use lots of different technologies to be able to create durable varieties. So I finally just wanted to have this slide here, which is essentially summarizing my talk. So in the face of a changing and complex pathogen population, we're trying to find new sources of resistance to warrior and then crossing these into our elite breeding pool, which takes time depending on the source of the resistance. We're also developing new markers in order to follow their inheritance and increase the frequency of these genes. And if we can increase the frequency, then this makes it much easier to make new combinations. And as has been mentioned, single genes are really not durable against the warrior race. So going forwards, we're making sure that genes are stacked together with other resistances so that we can maintain diversity and try and increase the durability of these varieties. Yeah, so thank you for your attention.
Thank you very much, Rachel, for that very clear explanation of the challenge facing breeders. Just a personal comment, it's quite encouraging, I think, that there has been good progress with varieties that do appear to have a bit more durability and probably more background resistance than previously. Mm-hmm. Now, I'll have a quick look at the questions here. I don't know if Catherine's been keeping an eye on it.
I haven't had any questions in for this talk yet, so if anyone's got one, please send them in.
There is a more general one about introducing new specific gene QTL controls to the yellow rust differential panel, but I think that's relevant to several of the previous speakers. So shall we hold that one back till the general questions at the end? Are you happy with that, Ruth, Rachel?
Yep, that's fine.
Yeah, I can't see anything that's specifically about the topic you were talking about, but there is time obviously at the end. If others have questions, can they keep them back till then? Okay, so many thanks.
Thank you.
At this point, we are actually more or less on time, which is quite good. It's again my pleasure to introduce Stuart Knight from NIAB. Stuart is actually Director of Crops and Agronomy at NIAB TAG, and I think he's a familiar face to many of us. So over to you, Stuart, to talk about integrated management of cereal rusts and mildews.
Thank you, John. I'm just going to get my screen to share, which it seems to be refusing to do at the moment.
We can see your screen, Stuart.
Yeah,
you can see it.
Stuart. Yeah.
Perfect. Perfect. Okay. Well, good afternoon, everyone. As is often the case with going last, some of what I've got in my presentation has been touched on by the other presenters. So hopefully what I'm going to say will be a useful recap, really, and perhaps a chance to pull it all together to a degree. So what I am going to cover, I'm going to say something a little bit about disease risk for the cereals, rusts and mildews, then touch on agronomic impacts, something around fungicide choice and timing, focusing there on wheat, and then finally some thoughts on what else we might do in terms of encouraging integrated disease management. So in terms of the factors affecting disease risk, that can be divided really into two. The first part of that really is about the risk of the crop being exposed to infection in the first place. And that's really a function of location, climate, your previous crop, cultivation method, sowing date, and also variety choice. And here I'm thinking about variety choice in surrounding fields and perhaps neighbouring farms. And then the second element really is the ability of the crop itself to resist or tolerate infection. And that's going to be driven by factors such as weather, variety choice. In this case, the variety you've chosen to grow in the particular field where you're trying to control the disease, sowing date, nitrogen management, soil fertility, and also general crop health. So all of those will come together to determine whether or not the crop you are growing this year is going to be at risk and need some sort of intervention. Dealing with the geographic disease risk, that one's relatively straightforward because it's generally reasonably predictable. These are some charts taken from the AHDB winter wheat into barley disease management guides from a few years ago, but they broadly show how the different disease risks work out across the UK. So yellow rust, I think, is as you all know, is pretty much an east-west split. So we have the east, which is higher risk, and pretty much the rest of the country, the rest of the UK, which is at moderate risk. Brown rust is more of a southeast northwest split, very high risk in the southeast, declining to low risk in the northwest and into Scotland. For winter barley, brown rust is a similar story to winter wheat, except we have some areas of the east coast of Scotland which are at moderate risk there of brown rust as well. And finally, mildew, where we have that greatest risk really across the north and northwest of England, rising up into the southern parts of Scotland, the rest of the country, again, pretty much at moderate risk. So those are sort of predictable factors that we'll all be aware of when in the farms that we're either looking after or advising on in terms of what we might expect in any given season. Of course, the weather has a big modifier on that. So again, climate will dictate perhaps the initial risk, but the actual weather in a given season will determine how problematic those diseases are and when they are a problem. So wheat, yellow rust, that's been touched on a little bit already, particularly in relation to the impacts of cooler weather, the damp spring weather, particularly overnight weather. But I think particularly, and I'll come back to this in a minute, the importance of frosts over the winter, and frosts particularly below -5 and the impact that has on survival of the disease through the winter. For brown rust, high temperatures in early summer really are the key indicator of a risk there. Some of you will remember back to 2006-2007 season, where we had brown rust coming into wheat crops very early. I can remember seeing it in crops at pre-stem extension in April, early April, and it really never disappeared from those crops. It just kept going right way through the season. So if it does come in very early and conditions are favourable, it can be pretty persistent. It's not just a high temperature disease, it also does need a high humidity or leaf wetness to trigger infection. Mildew, it's more a case of warm and humid conditions, not very particularly wet. On barley side, yellow rust, again, not a common disease by any means. I've only seen it 3 or 4 years out of my 30 years in agronomy, but it's favoured by really cool wet weather in the spring before fungicides are applied at the early timings. Brown rust, again, pretty much like wheat, warm and humid weather, especially during that sort of April to June period, the very critical growth period. And then mildew, again, warm, breezy conditions, short periods of high humidity, and it doesn't really like hot weather. It doesn't particularly like very heavy rain either. So some of those weather effects will also determine our disease risk and how we might need to respond. Just coming back to that question of air frosts, there was a nice bit of work that Pete Gladys did back in 2007, published in Annals of Applied Biology, which looked at the effect of a number of different parameters and factors on levels of yellow rust. And one of those was very clearly came out was the number of air frosts and the impact that has on the percentage of the crop that's diseased. You can see there, the more air frosts we have on my chart over the course of the winter months, the higher levels of infection. Sorry, the more frost we have, the lower levels of infection, beg your pardon. So if we look at how that's panned out in 2021, so this is last season, we can see this is data from the Met Office, where we are comparing the 2021 season with the 30-year average for the period before that. You can see that we started the year with a higher than normal number of frosts, pretty much across the whole country there. We were looking at the anomaly being anything between sort of 6-10 days more air frost than the 30-year average, so really quite frosty. But then we went into a period during February and March where the opposite was true. We had a distinct lack of air frosts, particularly in Scotland, as you can see, and in the North of England. And that was obviously good news from yellow rust point of view, less frosts, more chance for the disease to survive. But then a real frost bomb, for want of a better word, in April 2021, which I think helps very much to explain what Amelia was seeing in terms of the number of submissions to the UKCPVS yellow rust survey. We saw this very high level of frosts pretty much across the country, pretty much everywhere, and that certainly really put the dampers on disease in April and into May, which again contributed significantly, I suspect, to that reduction in the number of samples. This year, so far, we've had relative lack of frosts, again, particularly so in north of the border in Scotland, and particularly so through December. We have very few frosts. I haven't got the data up here for February yet. This presentation was just too early to capture that from the Met Office, but I suspect the number of frosts in February was also lower than average too. So I think the current risk for yellow rust is building up, running in the wrong direction as far as control is concerned, due to the lack of frosts. Now, quite a lot of the presentations this morning have touched on variety choice, so I'm not intending to go through this in any detail. You've heard a lot about varieties and disease resistance ratings already. But what I have done here is just put a very simple table together for the ratings for winter wheat, winter barley, spring barley, for yellow rust, brown rust, and mildew, whichever of those diseases are on the recommended lists for the 2022-2023 lists, and looked at the number of varieties there are in each grouping. So those groupings, if we look at those groupings, an 8 to 9 rating, obviously there are more resistant varieties. So those are varieties that we would hope in a typical season or in most seasons would not need specific fungicidal intervention in order to maintain control. We'd hope that disease resistance rating would be good enough not to require specific fungicide treatment. We might be putting fungicides on for other diseases, clearly, and they will be helping with the control, but we'd hope that overall the resistance ratings would be fairly robust. So variety ratings of 6 and 7, so those are the sort of more moderately susceptible varieties. For those, we are likely to have to use fungicides as part of the control strategy through the season, but there may be things we can do that would reduce the need for that. So obviously seasonal factors may impact on the need for that, but also some of our agronomic decisions may reduce the requirement for fungicides in those varieties as well, for specifically targeting those diseases. And finally, we have our varieties, our susceptible varieties, sort of 5 ratings or less. And for those, I think we have to accept that some sort of fungicidal intervention for those diseases is going to be quite likely in pretty much all seasons. But again, there may be things that we can do with our agronomic practices that can reduce the dependence on those fungicides, even though we may still need to use them. So what sort of agronomic impacts could we be looking at? So starting with the cultivation system, clearly, as has been mentioned already, the rust and mildew, biotrophic diseases, they can survive, they can persist on volunteers. And so anything that results in a high level of volunteers, that sort of green bridge between one crop and another, can mean a higher risk of those diseases. That's going to be perhaps most of a risk in successive wheat or barley crops and where we have used reduced tillage methods. However, I think it's very important to say that's not a reason not to use reduced tillage. I think reduced tillage has a lot going for it in many other respects, from the point of view of improving our soil health, and point of view of reducing our carbon emissions and various other factors, and ultimately economics as well. So I think where necessary, we can mitigate the potential increased risk of that reduced tillage by the other approaches that are there. Sowing date, again, often there's a perception that that early sowing is inevitably bad for disease and late sowing is always better, and that isn't really the case. Again, one thing that came out of the work that Pete Gladys did was to highlight that later sowing can exacerbate yellow rust levels in wheat. Now, that might sound a little bit counterintuitive, and it's not necessarily because the late sowing is favourable for the disease. I suspect it's at least in part due to the fact that later sown crops will be making that transition from juvenile susceptibility to adult plant resistance somewhat later in the spring, because they're a little bit behind in terms of the development, and that probably means they're more likely to catch weather in the spring that's favourable for yellow rust before they've made that transition to adult plant resistance. So I suspect there's an interaction going on there rather than just a disease effect on its own. Brown rust, like septoria, favoured by early sowing. Earlier we said more likely we are to have to perhaps tackle that in the spring, and as was highlighted in some of the sampling work for UKCPVS. Often we get samples for brown rust coming in pre-winter where the crops have been sown early. Mildew commonly develops on very early sown crops, but those very early sown crops often are slightly harder in terms of the crop growth. So actually the disease is more often damaging on later sown crops. It can be more difficult to manage in those situations. In terms of other agronomic impacts, if we have dense crops, they're very favourable for those rusts, and in particular mildew, especially where that density has been created by high seed rates, or indeed where we've got dense lush crops due to excessive nitrogen fertiliser or fertile sites. And not much chance probably of excessive nitrogen fertiliser in many farm crops this year, so perhaps that's a positive from a disease point of view, and perhaps a genuine effect might be seen where you are having to radically reduce your fertiliser rates this year. So that favours mildew, and I say lesser extent perhaps rusts. And finally, on other aspects of nutrition, micronutrients do play an important part in disease-resistant plants and in crop health generally. So micronutrients like boron, copper, manganese all have a role in various mechanisms of plant resistance and defense mechanisms. And we often see some interactions where a particular element is deficient, such as manganese deficiency can often lead to higher risk of mildew developing and becoming more difficult to manage. But I think it's very important to note that that bunging on lots and lots and lots of micronutrients does not get you out of the need to manage disease through other mechanisms. It's important not to have deficiency, but it's not going to solve all the problems. I think one thing I haven't put on there, which is probably going to be perhaps more relevant going forward too, is biostimulants. Some of the biostimulants that are available have claims about increasing the resistance of crops or their ability to withstand disease pressure, so their inherent resistance. So we could see some of those perhaps more in the future having a role to play. I want to move on to fungicide performance now and just show you some data from the wheat fungicide performance trials at AHDB funds, and focusing on yellow rust and brown rust in wheat. These are single spray trials where we test a range of products against those diseases as a single spray. In case of yellow rust, it's in a high-risk area in Kings Lynn at roughly at the T1 timing, and then brown rust, it's in Cambridge, again, high-risk area, applied more at the T2 timing, flag leaf timing. So we test a range of products from single actives, so azole actives like proline, Amarisa, Revysol, through and SDHIs such as Imtrex and Alatus Plus. And comment finally there astrobilurin, so older chemistry, and then mixture products, so the SDHI DMI mixtures like Ascaris Pro, Alatus Era, Revistar, and also now the QII DMI mix, which is Univoc. So we have a range of different products we're testing, both single actives and mixtures. I'm just going to show you two sets of charts. These are summary results from the last 3 years of trials from 2019 through to 2021. And these two slides there are two charts on each slide. It's the same set of data. It's simply divided between those two charts because there's too many curves to see on one chart otherwise. So on the bottom axis, the x-axis, we have the dose of fungicide as a percentage of the full label rate. Note these axes in this case only go up to 100%. I've taken out the parts of the curve that go between 100 and 200% because I think that's not a rate you can use in a practical situation. And on the y-axis, we have our percent disease. And so on the right-hand side, here we have our single actives. So these are straight SDHI products here and here, or straight azole products, or indeed straight strobilurin. Just a caution here, I've put it on my slide, although we test the products straight, it's very important not to use them in that way. Always use products in effective mixtures and programs, even if there are diseases like rust where currently we don't have a big fungicide resistance issues. It's still important to preserve that efficacy by using them in sensible programs and mixtures. And on the left-hand side, sorry, we have our mixture products. Now, the thing you will note is that actually those levels of efficacy are pretty similar across those products. There is a few differences between the curves, but in the grand scheme of things, they're all quite close. So the good news is we have a good range of products, both within the straight actives, but also within our mixture products, which have good efficacy against yellow rust. Now, you may say, well, the levels of control here are only fairly modest. We're only getting about 50–60% control at best. But remember, these are single sprays. applied at one timing, and we would not manage yellow rust in that way in the field. We would use a program of treatments, and that program would obviously draw together one or two or three applications of some of these products, or different products coming together in a program, and that would then give you much higher levels of control. But the good news is we have effective products and a good range of choices for yellow rust. Many of these things you'll be using for other diseases will be effective against yellow rust as well. Looking at the brown rust situation, again, similar scenario. Our straight products on the right-hand side here, our mixture products on the left-hand side. Again, our levels of disease on the y-axis and the percent of full label rate of the product on the x-axis. Here, the products draw apart a little bit more. We can see that in particular our SDHI products are Elatus Plus and Imtrex, which are the straights, and indeed our Ascarex Pro and Revistar XE, which are the mixture products, all very effective against brown rust, which is important given the slide I showed you earlier with the ratings on, highlighting that we have very few varieties with good brown rust resistance at the moment. They're pretty much all in the moderate or high risk category, and that presents a challenge, and we need the fungicides then to help support us with that. Some products though are not so effective, so Prylon a good fungicide for other things, but perhaps a weakness for brown rust. Again, important to consider the products and their strengths. So that's about the sort of efficacy of the products, but what about the timings and strategies? So for wheat, again, this was mentioned earlier, important to recognize that the latent periods differ between the different diseases we have. So Septoria is a relatively long latent period, anything between 14 and 28 days, depending on temperatures. Yellow rust shorter, around 10 days, but again, was mentioned earlier, it can survive perhaps more readily these days under higher temperatures, under which the latent period might be a bit shorter. And then brown rust and mildew, a relatively short latent period, can be as little as 4 to 5 days. And that affects how we need to manage those diseases. So what it means for yellow rust is that crops might be at risk before we apply our first T1 fungicide sprays, particularly if we have a winter where we have relatively few frosts and cold weather, and milder weather, which enables the disease to get going earlier in the spring. Or it also means that if our interval between our T1 and T2 sprays is too long, because we've gone very early with the T1 spray, or we're delayed by weather at T2, if we leave that interval too long, then the yellow rust can get going back in again in that middle period, and that might mean we have to intervene in that period. So compared to a conventional sort of T1, T2, T3 spray for wheat, those timings, we may need those additional sprays at T0 for mildew or for yellow rust, as I mentioned. We may need a holding spray at that period between the T1 and T2, something around growth stage 37 for yellow rust on those very sets of varieties if that interval between the two sprays is too long. Brown rust, because of its short latent period, can develop very rapidly after T2 and can continue to develop after T3 sprays have been applied and run out. But it's fair to say that very, very rarely do T4 sprays sort of applied at around 71, the cutoff for most fungicides. They're very rarely justified, very rarely economic. Just to highlight how that can come together in terms of the levels of yield response, this was a nice trial we did a couple of years ago here at NIAB, looking at a crop of JB Diego in Hampshire, where we had very high levels of yellow rust on the crop, untreated disease, roughly 58%. Yellow rust, a little bit of Septoria there as well. And we can see in that scenario that a single T2 spray only gave us about 1.5 tons. A T1 and T2 spray sequence added another 0.5 ton on from the T1 spray. Adding a T3 spray added another 0.5 ton again onto the yield response above that. And then having a T0 spray added a further 0.5 ton roughly on top of that response. So we almost doubled the response, really, going from a T2 only spray through to that 4 spray program. So in these seasons, in these situations where yellow rust is a very high, it's a very severe problem, we get high levels, you can see how each of those timings can contribute to the yield response that we get. For fungicide timings and strategies in barley, the key timings there obviously are T1 and T2, and the T1 often gives that bigger yield response in the north and west, or where Rhynchosporium and indeed in the context of today's discussion, mildew are higher risks. T2 often gives a bigger response in the east of the country, or where net blotch, brown rust, obviously in context of today's discussion, and indeed Remolaria are the bigger issues. T0 sprays can be needed if we have high levels of disease, active disease after the winter. So we come out of the winter with Rinco, maybe some net blotch, and perhaps more particularly some mildew, already getting going at high level. T0 intervention can help there. T3 sprays on barley, sort of, if can be useful, can be appropriate, especially where we've gone early with the T1 and T2 sprays to sort of fit with PGR timings. So if you're going on sort of growth stage 30, growth stage 39 to be able to combine with PGRs, then it's quite likely that the disease protection, particularly for brown rust and probably for net blotch, will have run out too early. And in that instance, a T3 spray can be a useful part of the strategy. But they're not always economic. And again, we don't really want to be too dependent on that approach. We should really be using those minimally as and when needed. Spring barley, key timings T1 and T2, and that should take care of the net blotch and mildew situation, noting that basically pretty much all varieties of spring barley have good mildew resistance. Unfortunately, they're all pretty weak on the brown rust, as my earlier slide highlighted. So what else can we do around integrated disease management? Well, there are a few things we could consider that's growing interest. One is variety blends. I know these have come and gone many times over the last few decades, but there is renewed interest, particularly in wheat. And there is some published evidence, not a lot, that shows that blends can reduce the delay development of mildew rust, especially in organic situations. But I think this is one of those areas probably that some further work is needed to see how useful that is in a conventional management situation. And secondly is intercropping, and that could work in a number of ways, either reducing the disease pressure through dilution, through having other crops in the same area of space, or acting potentially as a physical barrier, maybe to the spread of spores between one plant of wheat and another by having something else in the middle, or indeed by sort of direct inhibition, chemical inhibition, for example, of soil diseases. So again, there's been some work done on that. A nice paper from Zhang et al. In the European Journal of Plant Pathology, who looked at intercropping cereals with legumes and showed that this has the potential to reduce yellow rust and mildew levels, particularly in wheat and particularly during the early stages of epidemic. Again, perhaps helping to keep things under control before we go into other interventions. But of course, both variety blends and intercropping deliver some practical challenges, particularly in intercropping. And I guess we need to look more at how these can contribute to an integrated approach while still being practical and feasible. So just some conclusions to summarize, really. First thing, I think, in all situations, assess the potential disease risk. Look at where you are, look at what you're growing, look at how you're growing it. What does that mean for what you might need to do? Make good use of all the tools in the toolbox. Varieties, obviously, we've heard a lot about that today. Not just which variety do I grow, but what combination of varieties do I grow? It's very important not to go with all the eggs in one basket. Make use of good diversity as well as choosing things with good resistance ratings. In terms of general agronomy, there is no blueprint. It's all really about matching the situation, matching agronomy rather to the situation or the risk. So some circumstances it might be appropriate to sow early. Some cases it might be good to delay sowing. Some cases it might be a problem with if you are following another crop of cereal. Other cases that might not be an issue. So think about what you're doing and then match agronomy to that. And finally, the fungicide programs remain an important part of the program. We're really looking at reducing dependence rather than eliminating at the moment, and therefore choose appropriate products and use them at the appropriate timings, and then you'll get the best out of them. Just finally to finish, some acknowledgements really around particularly the fungicide performance work and the colleagues in AHDB, ADAS, SOUC, Harper Adams and Chuggas who are involved in that work. And that's it.
So many thanks there, Stuart, for a very interesting overview of all the options and kind of decisions that have to be taken. I often think, thank goodness I'm not actually a farmer, having to think of all these factors, but it's good that the information that's now available is much better than it was perhaps a few years ago. So I had a quick look at the questions panel. I'm not sure if there's any specifically for you, Stuart, so perhaps what we could do, as yours is a sort of more general overview, we could move on to the joint discussion. Are you happy with that, Stuart?
Absolutely, John. Yeah, go ahead.
Yeah, I think that might be a better way of dealing with it, because some of these questions are more generic and obviously you'll have a view on it. So at this point, I'm going to ask the speakers today to join us virtually, if they could, put their sound and cameras on. And I did actually ask Catherine Harries to help with kind of getting the questions organised. So I'm going to ask her first if she'd like to sort of kick off. Have you got stuff on your list, obviously, that we haven't covered?
Yep, so there's a couple of questions for Rachel that came in just after she finished the talk. So shall I go with them first? A question from Adrian Newton. Does partial or field resistance feature in your breeding strategy?
Okay, so slower pathogen growth, and these are often genes which on their own confer very limited effects on immunity. So we essentially need to stack lots of these genes together in order to achieve kind of an adequate level.
I think, Rachel, you've been speaking a little bit.
I guess
there.
some of the issues with partial resistance, although the idea is that they're likely to be more durable. Okay, I'll turn my camera off and see if that helps. Okay, yeah, so you're obviously needing to stack multiple resistances together to achieve an adequate level of disease. It's actually quite difficult to phenotype for partial resistance. So obviously their effects are masked by major genes. And then also, so one of the major kind of selections in the field in order to reduce numbers down within a breeding program is selections for disease, often diseases such as yellow rust. And essentially, you'll pick the most resistant varieties, and these are often varieties which have major genes. So these are taken through to the next stage of the breeding program. So yeah, I think the use is there, but it's difficult to put these in practice, particularly if you're having to stack 3 or 4 major genes together in order to get durable resistance, or what might be durable resistance in a variety. So if you're looking at major genes, how many genes do you, well, if you're looking at partial resistance, how many of these do you have to stack together, particularly to get a variety with a resistance rating of either 8 or 9?
Thank you.
We do have another one for Rachel as well. Shall I read that out now?
Yes, carry on.
So Lima grains seem to be already improving wheat resistance against yellow rust, and this includes the identification of the best combinations of R genes to use and development of genetic markers to trace those genes in the breeding germplasm. So I wonder whether you have any suggestions on what areas of research UK academics, geneticists and pathologists should put their main efforts in to help fight wheat yellow rust and other rusts.
Okay, so I think the work which has been undertaken by UK academics has really progressed resistance breeding. So, for example, characterizing different resistances and cloning genes. And I think kind of with the UKCPVS, we can monitor the pathogen population and we can monitor resistances within our breeding programs. I think one potential area which could be useful is gene editing. So, for example, we essentially want to be able to kind of move faster than the pathogen can. So if we can edit genes such as NLRs in vitro, then this might help us kind of keep one step ahead of the pathogen. And obviously, whilst we can't deploy these resistances at present, we really have to look to kind of the medium or long term with the deployment of gene-edited genes or varieties.
Okay, thank you. That question, I think, originated from Kostya Kanuka. There are a few that I've got on my list here. There was one from James Brown talking about the erosion of yellow rust resistance in some popular varieties. I think maybe that's directed partly towards Amelia, but others may have views on that. Paul Gosling did actually make a comment that some of the ratings have recently been adjusted anyhow to a slightly different scale than they used to be. So that might have affected that perception. But anyhow, would you like to comment on that to start with, Amelia?
So what was James Brown's specific question?
He said it was to do with the erosion. Let me find the actual question. These are my notes that I wrote from the question. You'll forgive me a moment. Yeah, he said, I have a question for Amelia about her slide showing erosion of yellow rust resistance in some popular varieties. Okay.
I don't think he actually got around to asking his question. I think he wanted to say it out loud, but he's now left. So
sorry, I missed that.
James, I think James wanted to say it out loud, but he's now left, so we can't answer that question if we don't know.
Oh, right. Okay. It's always a bit tricky managing the questions in this medium. Paul, let me just elaborate on what I said about Paul Gosling. He said there's an increasing number of recommended list wheat varieties with low powdery mildew resistance ratings. Is this related to the increasing frequency of virulence and isolates against many different resistance genes? Did you catch that, or would you like me to read it again?
We did actually answer that question at the end of the UKCPVS talks, and we answered that with the, to be quite honest, we haven't had that many wheat mildew isolates in, so our results probably aren't very indicative of what's out there in the present population, because like the last couple of years we didn't actually have any wheat mildew samples in. This year in 2021 we did, but basically I think we would need some more data to back up that with some more samples. Hopefully this year, if people can sample some more wheat mildew, we would really appreciate it.
We do have a question from Ruth Bryant from RIGC. So she says, are there any plans to introduce new specific gene or QTL controls to the yellow rust differential panel, especially those discovered in MAGIC and other NIAB lead projects?
So for the seedling virulence tests, at the moment there's no plans to add any extra varieties in, but it is something we review every year. So my seedling tests are getting bigger and bigger and bigger, and obviously the bigger the seedling test, it slows us down. But we do have quite a few additional varieties in there already that we don't always share the results with. So it is always something we could add in.
And then we've got one from Liam Wilkinson. So he says the yellow rust frost survival work, which was done on the warrior populations. This survival data is from 2007, which would be pre-warrior. Would this still be relevant, as we know warrior is a lot more robust?
So, I mean, I'm happy to say something about that. I think, yes, so the work that Pete Gladders reported obviously was from many years of work looking at yellow rust survival. I think I would argue the proof of the pudding is in the recent survey results, which showed that decline in survey samples, which at least indicates perhaps that that there is still a big impact on of frosts in terms of survival. But I don't know if any of the others have done that work specifically on yellow rust survival of the warrior race survival against frosts.
And then we've got one final question, I think, from the UKCPVS team. So for the wheat leaf rust adult plant resistance, why is 20 to 30% severity considered as a susceptible reaction?
So I missed that.
So
did everyone hear the question?
I'm going to repeat it. For the wheat leaf rust adult plant resistance, why is 20 to 30% severity considered as a susceptible reaction? Sarah, we can't hear you.
Sarah's still muted. I don't know if she's still muted by the organiser, but I can answer that question for her.
Thank you.
Okay, right. So basically, 20 to 30% in our wheat brown rust try was classed as susceptible because one, we calculate the sort of like the susceptibility based on that particular year's trial. So we might have had an overall lower infection level that particular year anyway. So if you average the results, then that would be considered susceptible. But what the main reason is because of the scale we use when we assess the plots is that 25% is actually half infected, half green. So the plot is literally half infected with brown rust and half it's clean. And for brown rust, that's actually quite a lot. That's a high level infection level. So 20% on a brown rust is a high level of infection. And we also have a very narrow window when we score brown rust. Sarah can only sometimes get maybe 3 assessments out before it all senesces. So it would actually, if the plant stayed greener for longer, it would actually, the levels would increase. So for brown rust, it's just that 20% is susceptible.
Thanks, Amelia. I did pick one up here from Nick Ballum. I don't know if others have seen this one. Have any significant yellow rust infection differences been seen between inoculated field tests and UTRL trials?
So Charlotte might have more information on this because I don't have a look at the untreated trial data. I just look at my inoculated trial data.
I don't have an answer at the moment. Okay, yeah, thanks, Rachel.
So I wasn't around for this.
Oh, sorry, Catherine.
I didn't do the RL work this last season. We do look at the comparison of the data between the natural and the inoculated trials. If there's a big difference between the ratings produced from each of those trials, then we'll look into it and make sure that that's definitely the answer and investigate it. Why there's such a big difference? I'm afraid I can't comment on what the data look like this year, but we do look at it.
Thank you, Catherine. I think that's pretty much most of what I've seen on my list. There was a question about the frequency of MLO 12 in UK barley varieties. Quite a specialised question. Does anyone know the answer to that?
MLO 11.
Oh, is it MLO 11? Sorry, my writing's not very good. Any comments on that? No. Okay. So we don't have.
What was the specific question?
It was
I wonder how frequent is MLO 11 amongst the current UK barley varieties. I think all the ones on the recommended list do have an MLO gene. I don't know which one it is, but that's why they've all got such complete resistance to powdery mildew at the moment.
Okay. So we know that that's a durable source on the whole. Okay, well, I think unless Catherine's seen anything else, I think that's most of the questions in the panel on the actual questions list dealt. I think I should give an opportunity to the speakers to ask their own questions at this point, having sort of participated in this event. There might be additional comments you want to make or questions you want to ask the other speakers. Anyone volunteering at this point?
I suppose I'll pose a question, and that is, I think if we look at the disease resistance ratings, we seem to sort of swing from doing really, really well on one disease and really bad on another disease, and then over the next decade, it kind of shifts around. So are we achieving a good balance of resistances across the varieties we have, or are we tending to sort of focus too much on one thing and then something else slips, and then we have to spend the next 5 or 10 years then trying to rectify that?
That seems to me a very fair question. Does anyone have a particular viewpoint on it? I mean, my own contribution to that would be And obviously I'm not a breeder, and I'm not up to date perhaps with all of the factors involved, but seems to me that some of the more dramatic failures of resistance that we saw in the not too distant past aren't now happening to the same extent, but we still have problems with erosion of resistance. And presumably that is because, I mean, this is just me flying a kite. It is because we've got more background resistance in those varieties, and the breeders have paid more attention to that. So in that sense, I think we're moving in the right direction, but it doesn't answer Stuart's specific question, which is, are we focusing too much on septoria or whatever the current top of the list priority is? Maybe it's black stem after Diana's talk.
Perhaps I could, just give I think it really depends kind of on the incidence of the disease that you see in your nursery. So, for example, if has been talked about, if you don't get very much wheat mildew, then it's very difficult to select for resistance or susceptibility to wheat mildew. And I guess with particularly given the strong environmental influence on disease, that it can be very difficult. So other than last year, 2016 was really the biggest septoria year. So I think it's really dependent on being able to capitalize on when you have high levels of disease, and then being able to kind of use that information. But yeah, I do think it does make it difficult, particularly for things such as mildew or for example fusarium, where the environmental influence is so great, which means that you can't, for example, collect data every year like yellow rust, which is perhaps why we have such a wealth of information and a focus on yellow rust.
Thank you, Rachel. Any other comments on this?
I guess a related challenge then, should we be worried about the fact that all our spring barley varieties on the recommended list are rated 5 or less for brown rust, or is it not a concern because we simply don't think brown rust is a big enough problem, because we know it's not a major issue and it can be readily controlled with fungicides?
Any views from the survey team on that?
I recognise it's not actually covered under UKCPVS because the barley work focuses on mildew, but just a more general challenge, really.
I guess, well, your presentation showed that we should be diversifying our toolbox. So yeah, maybe it is an issue that we don't have some stronger resistances in the spring wheat. We shouldn't just rely on fungicides. But maybe, as Rachel said about priorities or disease pressures, perhaps, yeah, it's not a priority.
Okay, so we do have a new question here on the question list, which starts off, thank you for all these nice presentations. I have 2 questions for Dr. Diane about stem rust scoring. Hang on a moment, that's just slipped up the panel a bit. Stem rust scoring at adult stage in the field. How many times you score the field trial? My second question, well, let's do the first one first. This is from Vided Abadai. I'm sorry if I've misspelt your name. Sorry, mispronounced your name.
Yeah, so we did the field trials also over multiple seasons. So it wasn't just one season's scoring. So it was scoring over 3 different seasons. We did them as adult plants in the field, and also we did do some limited glasshouse experiments on both seedlings and adults using the isolates that were present in South Africa. So I think it was quite robust, the scoring system, and it's done by those who score readily for all the companies as well in that region. So yeah, I think it was unfortunate that we didn't really identify much resistance among the varieties that we scored. They were all in the susceptible range.
Okay, and the second question is about stem rust re-emergence in countries where barberry trees are not found. how much distances could uridinospores travel?
Well, they can travel great distance between continents. So, but it's about the level of infection that they would induce. So if you didn't have any local inoculum and it had to arrive into the country, then it's about, you know, how far would they have to travel and how many would reach the region that you're talking about. There are countries in Europe that were very effective in removing Barley. So Denmark is one of them. They were really good at making sure it was eradicated. And it's hard to actually find Barley, or at least common Barley, in Denmark now. But other countries like the UK, because it was more farmers driving the removal, a lot was left behind in certain regions, and these bushes make lovely, beautiful red berries that the birds love to eat and spread around. So they, of course, have naturally increased in prevalence.
Okay, thank you, Diane. Right at the last minute, Adrian's managed to ask another question. Adrian Newton, why don't we have a problem with yellow rust on barley? There's a brain teaser for you all.
Well, can I just say, from our inoculated field trials that we used to run, it was quite hard to get yellow rust of barley going in the field, even when it was heavily inoculated, and we ended up having to mist it to create, because it likes quite wet conditions. So maybe it's just that now it has reached such a low incidence in the UK. Over the past, decade, we've seen fewer and fewer samples sent in, and incidence was getting less and less. So I think it's probably maybe due to the weather. A lot of it, it's just
maybe an example of climate change actually having a good effect.
Yeah.
And because it was getting of less importance, we stopped doing recommended list ratings for it. And in the first year that there was yellow rust recorded in barley, maybe that's the way to get a disease to come back, is to stop doing ratings for it.
I think practically in the field, we tend to find that usually we have put the first fungicide onto barley before it starts to get going. So I can remember seeing it a couple of times, as I say, 2 or 3 times over my 30 years, and it's almost invariably been where it's come in before we've put our first fungicides on, because it's come in very early. We've had favourable weather very early. Most times I think we get the fungicides on the first trays before the disease, the conditions are right for disease, and then we don't end up with any disease, because most of the fungicides we use for other diseases are very effective at keeping yellow rust out in barley. There is no difficulty controlling it with what the products we have.
Thank you very much, Stuart. I think we've now reached the end of our allotted time. I'd just make a general comment. Quite a few people have questioned about the availability of basis points for this. So is there someone who can answer that? I'm not involved in that. Does this rank in basis points?
I think if you send your basis details to Maya, who organised the event, then she'll get them added for you. So
they should be directed to Maya at AHDB. Yeah. Okay. Okay, so if there's nothing else, I think it's just down to me to really say thank you to all the speakers for producing such an interesting mixture of topics, ranging across a wide range of currently important issues. Thank you to all those who've attended. Unfortunately, we can't see you all. Apologies for some of the relatively minor technical glitches that we experienced. I'm afraid these online meetings are always a bit more tricky than the real thing, in my view. Hopefully it didn't cause too much disruption to the actual amounts of information that you received. And I should perhaps thank those who organised this meeting in the first place and helped us to get our computers running in the correct way some of the time. Okay, so thanks very much for your attention. At this point, I think we'll draw the meeting to a close, and I hope you all have a very successful growing season, those among you who actually do the serious business of producing our food in the first place. So thank you. We can all log off.